Bon. Alors, ça, this is the first atelier that we are organizing here. We try to think about a topic that would be interesting for the people that actually comes usually in this laboratory, to this platform. So the idea came at a certain moment when Stefano wrote me that with this idea of organizing something here in Paris. And Stefano Tortora is someone that has a lot of experience on EEG. I think he will show you something about his works, his own works. He participated, actually, he won a few times the Cibathlon. Is a kind of athletic competition for people with several different disorders, in particular focusing on the brain-computer interfaces for controlling a wheelchair. Okay, so the idea was, okay, maybe here, you know, at LUTIN we have several EEG platforms, and we also had some discussion with some of you, maybe Julie, maybe about the possibility of using it. And I always said, yes, you can use it, but it's complicated then. It's not just a problem of acquisition, it's also a huge, I mean, there is the entry point is not so straightforward. So with Stefan, I said, okay, maybe we can just make a kind of tutorial, a kind of hands-on, focusing on how to acquire, how to elaborate EEG practically. You will see, usually we have a different, okay, you will program, okay. So it's something, it will be something practical. You will use Python mainly. Actually, there are two school of thought, you know, few people that actually works with this using Python, people that work with this using MATLAB. I take the responsibility of choosing Python because I think in some way is a language that is widely used, in particular right now with all the spread of AI, you know. So I think okay, I think do we have a planning? Because at some moment we make okay, you have a plan. Okay, but listen, I leave the floor to Stefano, and I think I hope you will enjoy it because I mean, I repeat, we are trying to do our best, but this is our first time. Okay. Thank you.
Thank you, Salvatore. So, good morning, everybody. So, the idea of what I will present you is really to be something informal. So, to provide you with like a sort of a tutorial. There will be something to program, but not much. So, I prepared for you a lot of material so that it could be basically an entry point for you, so that you can take the examples that I will show and we can discuss together during the discourse. But then you can also reuse or readapt to your specific scenario. So I try to give really a lot of examples that we will go through. We will first have, of course, a more theoretical part, just so that we all start from the same theoretical points with respect to EEG signals. And then we will end, particularly in the afternoon, there will be more the hands-on activities, which again will be an idea of exploring together some libraries and some tools provided by Python for working with EEG. Since I know that you mostly focus on neuropsychology and human-robot interaction, most of the examples will be actually focused on EEG analysis and processing for offline applications, so to extract the useful data from the EEG signals. And then at the very end, if there remains time, I also provide you with an example of a pipeline for a brain-computer interface similar to the ones that we use currently in our research activity. But let's see. So, but I want really to keep it very informal. So be free to interrupt me, say any comments if you have any problems. So it will be very interactive. So I don't want, I prepared the presentation just to follow it so that I have an order, but I want this to be like a straight presentation where I just say everything and you just listen. So I'll try my best. It's actually also for me the first time that I try to do something similar. So let's see what happens. So just a very short introduction on where I come from. I'm assistant professor in the University of Padua, which is the northern part of Italy, and in particular in the Department of Information Engineering. So I am a computer engineer. Actually, I come primarily in my very first experience was in biomedical engineering for a bachelor and master, and then I moved to computer engineering for exploring more the human-robot interfaces, and then the field, my current field of research, which is neurorobotics. Actually, within the Department of Information Engineering, I belong to a big research group which belongs to the Intelligent Autonomous Systems Laboratory, which actually consists of 6 faculty members covering in different areas and aspects of intelligent agents and intelligent robotics. So from, like say, the very first area that were born in this laboratory were, of course, related to mobile robotics, industrial robotics, and then more recently, robotic vision and machine intelligence. And when I started my PhD in Padova, we also started like a new branch within this laboratory, which is the neurorobotics branch, which the main focus is to exploit both let's say the knowledge is in processing and machine learning with the artificial intelligence applied to robotics to create a symbiotic system that combines both the intelligence of the robot and the neural interfaces. We are actually quite a huge group with more than 20 PhD students over the different areas, and more than 30 between master and bachelor students. And we also have a lot of people coming from abroad for the visiting period, both master for the internship or thesis, and also PhD students for the mobility period. So feel free to, if you are interested or if you see any contact point, to contact me, and we are very open to collaboration on any topic related to robotics and human-robot interaction in general. So together with let's say my affiliation to the Intelligent Autonomous System Laboratory, we actually extended our collaboration to create a more interdisciplinary research group, because as you may understand, working with EEG and robotics, it requires a lot of like different knowledge. So we actually created a robotics and neuroscience group, which fuses together, let's say, engineers, biomedical engineers, control system engineers, computer engineers, of course, and neuroscientists. So we have also activated a strong collaborations with neuropsychologists, physiotherapists, and of course physicians. Okay, so we try, so as we believe that it is the best way to work in this area. So just very short introduction on what I am. So now let's move directly to the topic of the day, which is the workshop. So as you may have read in the workshop description, the idea is to offer you a comprehensive introduction on the fundamentals of the EEG signals, in particular how to we'll start from a pre-recorded dataset, of course, as you will not have the possibility to record data, but starting from different type of data, so also different type of data formats, so that you can, let's say, understand how to work with different data formats. The idea is to give you the basics of EEG signal processing, cleaning, extracting features, extracting the main information that you would like to analyze in different EEG experiments, covering both the theoretical part and the practical aspects. So the idea is that by the end of this day, you will learn, of course, what is EEG, and particularly its neurophysiological origin, which are fundamental to understand the limitations and the problematics that arouse when you start working with this very difficult signal. I work with really a lot in signal processing, not only EEG, but also energy, speech, kinematics, force data, and among all of them, EEG is clearly the most complicated one. So beware if you start working on it, because it's super fascinating, but also there are some, let's say, criticalities and difficulties that must be taken into account. Then some state-of-the-art EEG processing methods, and I hope to finish this part in the morning, and then leave the afternoon for focusing more on the main analysis that you want to do in different EEG experiments related to cognitive neuroscience. And then in the final part, just basic concepts on brain-computer interface. Again, if it remains time. So let's see. Let's see. This is roughly the program that I imagine. So with the idea of having this like very first part on welcome introduction and a theoretical part on neurophysiological basis and EEG recording, and of course some coffee breaks in between because it's going to be a very long day for all of us. And then we will start immediately after, hopefully before the lunch break, already working a bit on the practical part, at least for processing, data visualization, and everything. Then we will have a lunch break. I expect it to have 1 hour and a half for lunch break, but we can reorganize it if we want to make it shorter or longer. I mean, we can decide. This is just a suggestion. And then in the afternoon, we will have, as I said, the practical parts on different type of paradigms, and concluded with some discussion and closing remarks, particularly if you have any doubt or if you have any other curiosities or we want to explore other thing. I'll try to give space also for that. Okay, so first of all, since I mean now there's going to be this theoretical part, I prepare for you all the materials for working in Python that you can find at this link. So I leave it here, where you can find not only the slides that I will present, so that it remains for you with all the theoretical information, but also a zip folder that I would suggest you to start downloading it now because it is a bit heavy, as it also contains some data. And so my suggestion is to download the zip folder, and once it's finished, to put it, for example, on your desktop and decompress it so that it's a blank folder. This is the, let's say, the so-called Python environment that contains all the data and all the scripts that I prepare for you, and we will reuse them in the hands-on part. We need the permission to the.
Yeah.
Right. Why is that?
Blocca la condivisione dello schermo.
Oh. Try now. Otherwise, I'll just give you access manually. It's okay. Work. Okay, perfect. So you see here the slide. Here you can download this Paris Workshop EEG zip that contains basically is a copy of this folder here. Okay, which contains the Python files that we will use later on, and the virtual environment and the data. Okay. So you can just put it in your desktop and with right click uncompress, and you can open the.
Puoi rimettere il link.
Yes. Here we go. Okay, so this is the link. Well, then, so one thing is, I don't know if you started working on the email that you should have received from Salvatore regarding the installation or maybe of Jupyter or something. If you didn't do it, it's not a problem. I prepared like a very short tutorial on how to let's say set up your Python environment so that it could be useful also if you have to work on it and how to connect the virtual environment to Jupyter. If you are already using Visual Studio Code, that would be the easiest way to work with the environment. But if you don't, I will show you an example on how to connect it to Jupyter Notebook. I would not suggest to use Colab instead, because it's a little bit more complicated than to connect the virtual environment to Google Colab. So if it is useful, then it's going to take a lot of time just to reorganize. But with Jupyter, it's very simple to use and to connect. So we can follow it step by step. Okay, you all have the link. Can I move further? Okay, So as you see, it will take a bit for the downloading and compressing. So in the meanwhile, we can move on with our presentation outline, starting from the first part, which is the neurophysiological basis of EEG recording and technology. So first of all, what is EEG? EEG stands for electroencephalogram, as you may probably know, which is probably the most well-known non-invasive technique that is used to record the electrical activity that is produced by our brain when the neurons within the brain fires. And so they communicate, as we'll see better later, through basically neurotransmitters, because the communication of these neurotransmitters, it can be seen as an electrical communication within our brain. And this electrical communication allows us to record at least some activity of this by placing electrodes on the scalp. So if you try, or if you ever tried to record the EEG signal, normally what you end up with is something like time courses similar to this, where you can see it is quite a noisy signal, which is not that easy to identify elements in it. And they also have several components, because you see there are these low waves and high frequency oscillations. So it is very complicated and heterogeneous signals, which can really be studied from different perspectives. What maybe we can directly see visually, and that is what normally is done by since years by neuroscientists and physicians in the clinics, is to look to, let's say, main components or main visual components of the EEG signals. So one thing that we can immediately notice here is this huge peak that appears on basically all of these, which are different channels. So basically different electrodes placed in different parts of the brain. Okay, each of them recording an electrical signal. Electrical signal whose let's say range is very small. Normally, like a single signal is in the order of 10 microvolts, probably lower. And so it's a very, very tiny signal from which we need to extract the information. And the main problem is that on top of these signals, which is already very small, there are this type of activity, which are, as you will see better later, are artifacts. So are unuseful information generally for us. So we don't really want to see blinks in our EEG signal. But as you can see, the effect of a blink is quite important in the EEG. So we will have to take care of it. Nevertheless, when there are maybe not this much of artifacts in our signal, we can also see other components which are, let's say, clearly visible, which are these oscillations that you can see here, but also here, that seems at a very precise frequency. And we will see that this is a very specific oscillation in the EEG signals, which is an oscillation at 10 Hz. So normally the kind of information that we can extract from this signal are either like time peaks in very specific moments. These are normally related to artifacts, but not only, or oscillations. So that means we can see that the EEG is modulated at a very specific frequency. In this case, a half oscillation at 10 Hz, which is normally the best visible one. But not only, so we can also see that in other signals, we can also have other type of oscillation at other type of frequency. So it's actually a more complicated signal than this. The EEG has actually quite a long history. It was first measured in 1924 by Hans Berger, which actually it was the first one recording EEG signals, but it was a little known in history, particularly in the literature. Even only, let's say, its work was mostly discovered when it was used by the Nobel Prize Edgar Adrian for his research in physiology, and he cited the work of Berger on the EEG recording, and there were all the, let's say, the neuroscientific literature using EEG starts from. And the first thing that was firstly analyzed ever in the EEG signal is exactly these components at 10 Hz that was clearly visible also at that time, even without any computers, even without any specific signal processing technique. This oscillation at 10 Hz were clearly visible. So they were the first one studied also in 1924, and then replicated by Edgar Aden. So to understand better where the EEG signal comes from and why it has these characteristics, let's just have a brief look of how the brain is organized, so that we can understand, let's say, the meaning of the different parts. So the EEG signal originates mostly by the cortex, and the cortex is organized in different areas, which are normally associated to different functionalities. So, for example, we have the so-called frontal lobe, which is basically this frontal part, which is associated, let's say, in neuroscience, but it is mostly, let's say, a convention. But it's thought to be associated to the part of decision making, motor planning, and problem solving. Then we have the parietal lobe, which is basically this part of your brain, which is related to spatial processing, all the mechanism of attention, and also some part of sensory integration. So there is also like a sensory area within your parietal lobe. And actually, in between the frontal and the parietal lobe, we have another area, which is the motor area, which is kind of overlapped between the frontal and the parietal, but it's a very specific area, which is related to, again, sensory integration and motor planning, but specifically for motor movements. So it's in charge of all the sensory motor parts, and is particularly important in EEG signal, and particularly for brain-computer interface. See it better later. So we have the frontal lobe, parietal lobe, then the temporal lobe, which is this part here, which is mostly related to memory and language processing. Sometimes it is also possible to extract information related to the auditory activity, but it's a difficult area to process due to its vicinity to the neck. So it produces a lot of noise from the neck muscles. But if you are working in language, for example, we started recently a project on speech decoding using EEG. And for that, we need to extract let's say the area related to language. So we are starting now working also on the signal produced by the temporal lobe. And finally, the occipital lobe, which is to the back here, and it is related mostly to visual processing. Okay, so analyzing images and the data recorded by our eyes. Okay, so this is a very say general and schematic representation of how our brain works. Actually, it doesn't work like that. So it was very well discovered, not even recently, that actually this was more like of a convention from the research in neuroscience that required like a sort of categorization and classification of different brain areas. But actually, there are networks that are spread all over our brain, and they are all responsible for all these tasks altogether. But mainly, we can say that the main networks responsible for visual processing are in the occipital area. The main networks for the parietal lobe, for the attention, and the parietal lobe, and so on and so forth. So there is a sort of clustering of networks, but it's not like a strict separation. So given this organization, which is, let's say, a spatial organization of the EEG, let's get deeper into the brain to understand better what is happening and why we can see the EEG signal. So the cortex is basically organized in two main parts, which are the gray matter and the white matter. And what we are interested in when we talk about EEG signal is the gray matter, which is the external part of the brain, in which if we zoom in this part, we can see that in this gray matter we have the neurons which are going perpendicular to the cortex. Okay, and these are spread all over the surface of the brain, which is actually very extended, as these as you may know. Actually, the brain is organized in sulcus and gyros, so that they maximize the amount of surface available in the brain in a very limited volume. Okay, so in all this surface we have these perpendicular neurons, that goes out of the brain, and these are the main neurons or the same main part responsible for the EEG generation. If we go deeper again, and we check exactly what is happening in that area, that now we will call it the neocortex layer, because it basically was the latest part of the brain to be to be generated during the evolution. So that's why it's called the neocortex. So we have this vertical organization of neurons that actually go through different layers from the surface to more deeper layers in the brain. And this organization is actually present in all the areas that are shown above. So the prefrontal, the primary motor cortex, the parietal association cortex, and the visual cortex. They all share exactly this type of layer organization with these vertical neurons. And among all of them, let's say that the main ones responsible for the EEG generation are the pyramidal layer. So the pyramidal cells present in the fifth layer. Okay, so the EEG has been discovered to be generated exactly by these neurons, which are the biggest one, and thanks to their orientation, are the ones mostly responsible for generating the electrical current that then we measure with the EEG electrodes. So how does this happen? Basically, we have the cortical surface. We have all these an example of the fifth layer, and so we have all these pyramidal neurons in the vertical, in perpendicular direction, actually in perpendicular direction to the cortex. Okay, spread all over the cortical area, and the reason why they are mostly responsible for the EEG signal generation, as we'll see better in the next slides, is that they have a very interesting characteristic of firing in synchrony. So they are very synchronous neurons, and so the overall activity of all these neurons together is what generates the EEG signal. Not only, the other characteristics that they have from the neurophysiology is that with respect to other type of neurons, the potential generated by the activity is longer than in other cells. This is not mostly due to the size of their cell body. And so since it is a long-lasting potential and we have a lot of neurons firing together, they are the main components producing the EEG signal. So there are also the effect of other neurons, of course, but these are the main ones involved when we record the EEG signal. So basically, this is exactly the idea. So as you probably may know, neurons are very specialized cells for electrical communication, in which they have like a receptive part, which is composed by dendrites that collect information from all the other neurons that are connected to our neuron, that then integrates this information in the cell body. When this information surpasses a certain threshold, we have the generation of an action potential. Action potential can be seen as a spike, as a spiking activity. Okay, that once it is generated in the cell body, then it propagates all over the axon. It arrives to the synapses, and then it is propagated to the other neurons that they receive from the dendrites, and so on and so forth. And so we have thousands and thousands of neurons in our brain, that all working in this way. And so what we do measure effectively, if we focus inside our brain, we imagine to just think about take like a slide of the neocortex, particularly focused on the fifth layer. So here you have your pyramidal cell that is receiving information from the other connected pyramidal cells that you can see here. Okay, when they receive enough information to pass the threshold. Okay, so this is the signal that I could record if I would be able to insert a needle exactly inside that neuron. What I would measure is exactly this type of signal. So here is accumulating some current coming from the other neurons. When this passes a certain threshold, then you have the generation of normally a train of action potential, which are exactly all of these spikes. Okay, and then there is another resting period, and then another train of spikes, and so on and so forth. Okay, so this is what happens inside the cell. And these are the spikes that propagates also to other neurons. The problem is that when there is this communication between different neurons, this communication is not perfect. So there are inefficiency, as in all the system, in no communication system, there are inefficiency. So that means there is a leakage of currents between one neuron and another. So some of the neurotransmitters that should move from the synapses to the dendrites of the other neuron are dispersed in the extracellular space. This dispersion of neurotransmitter in the extracellular space is exactly what it causes the generation of what is called a primary intracellular current, okay, which is basically generation of the primary intracellular current is what is obtained by the activity within the neuron. Okay, so it's basically a magnetic field that is generated by the passing of the action potential along the axon. Okay, the dispersion of the neurotransmitter in the extracellular space is what it generates the secondary volume current. And it's exactly the secondary volume current that is what we measure with the EEG signal if we place an electrode on top of the skull. So basically, once there is this communication, part of this electrical signal is dispersed and it propagates in the extracellular space. It arrives to the scalp, and when it arrives to the scalp, we can record it with the electrodes. So that means that actually, when we record the electric activity through the EEG electrodes, what we are recording is not the signal that is communicated between the different neurons, but is actually the dispersion, the information that is dispersed and not used by our brain. So I always because I think is like a powerful example to say that when we work with the EEG signal, what we are really measuring is the noise produced by our brain when we think. So it's not really what our brain is doing, but when it's doing our thing, it's producing some noise, and what we can record is exactly this noise. And we need to work with what we have. So exactly this noisy information that is still very important. Okay, so we have to be aware of this, that what we are really measuring is not this electrical activity that is exactly related to the spiking of the neurons, but is actually what it remains as what is not transmitted in this signal. Okay, so it's again the noise. Okay, and normally this field potential that we measure through the EEG channels is in a frequency content which is lower than 100 Hertz. So it's difficult to find something at a higher frequency, and this is normally due to this layer. So when we have this electrical activity, that it can also be in a very high frequency because it's the frequency of the neuron spiking, which is spiking at a very, very high frequency. But actually, when the signal propagates, then it encounters this layer, which can be seen basically as an isolating layer. It's our scalp, our skin, the dura mater, they work like a filter. Basically what it does is that when this high frequency signal passes this layer, it is filtered with the high frequency component, which is the smaller ones. So what we received through the EEG electrode is actually the low frequency part of our brain activity. So not only it is a noise, but it's a filter noise. So it is even more complicated than this. So these are, let's say, knowing how the EEG is generated from firing neurons. It is important because we need to know which are the limitations. What can we really do with the EEG signal? This was the case of a single neuron, and it would be really okay if it is still the noise of a single neuron. Fine, I can work with it, but at least I know how the single neuron works. I'm okay. But it's actually not even like that. We cannot record the activity of the single neuron with the EEG. What we actually record is the activity of a population of neurons. Okay, so if we imagine to have several neurons within our brain, all of them communicating through spikes. Okay, what we really see is basically the convolution, so the union of the activity of all these neurons that are collected by the single EEG electrodes. So our EEG oscillations, they come from let's say the combinative potentials generated by different neurons within our brain. Nevertheless, this can still be very informative. For example, if we have a situation in which we have all our neurons that we know they are pyramidal cells, they are able to work in synchrony, and they work perfectly in synchrony. What it happens is that what I see at the level of the EEG is an increase of amplitude. So when I see an increase of EEG amplitude, so an increase of the EEG signal, this means that the neurons that are generating this signal are firing in synchrony. This is the reason why we will call it also later a EEG synchronization, because it exactly comes from this synchronization of the activity of the pyramidal neurons. On the other hand, when we have our neurons that starts not working in synchrony, for example, we have some neurons firing at a very high frequency, some other neurons instead firing at a low frequency, or even firing at the same frequency but not phase locked. Okay, so they are not locked in time. What we see at the level of the EEG activity is a reduction in amplitude, and we call this reduction in amplitude EEG desynchronization. At the very end, what the EEG signal actually look like is something like this. Okay, so what I showed you before is actually would be again the ideal part that I just manually draw that I expect to have, but things are not that easy. So a real EEG signal is something like this, in which you have normally a lot of EEG channels because you want to cover all of your brain, and all of these EEG channels produce this very noisy activity, which actually have a very tiny, which the oscillation has a tiny amplitude. And then we also have this, as you can see here, this high amplitude, low frequency waves, which are normally not very informative. Okay, so and you can see here also we have here a certain amplitude. Here we have another, like seen like this is not very easy to extract any information at all. So the raw EEG signal on its own with respect to other type of signals, such as I don't know if you ever worked with EMG or if you ever worked with the speech production. If you see this kind of signal, or even the signal produced by a gyroscope, they are immediately clear, even if you don't do anything. I mean, you just record the signal, you look at them and say, okay, I understand that here there's a movement, here there's not, here there's an activity, here there's no activity. In the EEG signal, this is not possible. So there's no way you can extract information only by looking to the raw EEG. So we need a lot of processing to make this signal useful, and that's what we'll try to do in this lecture. So one of the first things that's been done in the neuroscientific literature is not to study the EEG signals in a row, so as it is, but to try to identify the main oscillations that are present within our brain. So what they end up with are the so-called EEG rhythms. EEG rhythms are nothing else than the raw EEG signal that I showed before, filtered in very specific frequencies. Okay, so for example, when they filter the EEG below 4 Hz, so they take only these low these slow components, okay, these ones that you see here, okay, they are also called delta rhythms, which are below 4 Hz, and has been shown in the neuroscientific literature to be related to, for example, sleep, your sleep activity. So it can be used, for example, to measure, to understand if you are sleeping well or not well. So there are some studies on them, but in general are the waves that are that increase their amplitude when you are sleeping. If we increase in the frequency domain, we have the theta rhythms between 4 to 7 Hz, which are instead related to drowsiness, or in general or it can also be associated to effort. So when you are relaxed, when you are maybe a little bit sleepy, or if you're not doing anything particularly effort, then you have an increase of this theta activity. When instead you are concentrated or you are stressed, you have a decrease of this theta activity. So it can be associated to this type of, let's say, behavioral aspect. More interesting actually are these ones, that the alpha waves, which I said that is at 10 Hz. Actually, it's not necessarily at 10 Hz. The alpha is a rhythm, so it's between a certain frequency range, normally between 8 to 15, 8 to 13 Hz. It may depend. And this is as I said at the very beginning, is the main oscillations of the EEG signal. So the ones that you can see also in terms of size is the bigger one, so it's the easier to see. Yes.
You just said it depends whether it's 13 or 15. It depends on the person, or it depends on
It depends on the
individual?
person. So each person has its own alpha peak.
Mhm.
So I could have an alpha peak at 10 hertz. You could have it at 12 hertz. Someone else. Normally, if I then I will show you better in the slide, still there is a peak covering a certain band,
Mhm.
and this is exactly this band. But the peak itself can be shifted a little between these range. And normally there are also other studies that associated the position of this peak, for example, to age. There are children normally have this peak at a higher frequency, so closer to 15 hertz, while elderly have this frequency closer to the low frequency, closer to 8 hertz. But again, in grand average, so you may find an elderly subject, one with 15 hertz, one with 8 hertz, and on average, they are slower than the children. But so it is a person specific. And these alpha waves are very interesting because they can easily be modulated through attention processes. Actually, there is, I mean, if you are able to record the EEG activity on yourself, one of the first things that you do to check that you are really recording an EEG signal, a proper EEG signal, and not just noise, is to see the signal while the subject has his eyes open, and then to ask the subject to close their eyes. And you immediately see that when he's closing his eyes, you will see immediately an increase of this exactly of the EEG signal will exactly look like this. So all these components will basically disappear, and you will see for certain periods exactly these waves. And this is due to the fact that the alpha rhythms is related to relaxation, inhibition, and attention. So if you are relaxed, you have an increase. So if you're if your brain is relaxed, you have an increase of the alpha amplitude. If you are concentrated, you have a decrease. And the reason why you have this huge variation when you open and closing your eyes is due to the fact that the main effort that your brain does is actually to process vision. Processing images is the most complicated thing that our brain can do. And so as soon as you close your eyes, immediately your brain says, oh, finally, I can relax. You relax and increase its alpha peak. Okay. So this is the only component of the EEG signal that you can really see visually, even without any specific processing. Okay, and it's also interesting it sometimes happens, particularly with a subject that has this peak very high, that you ask them to close their eyes, and then when they have their eye closed, you will see the increase the alpha amplitude, and then you ask them to perform calculations, like complex calculation, complex multiplications, or I don't know, whatever. And you immediately see that this peak reduced. This is immediately reduced. Why? Because okay, they relax their brain, but then you ask the brain to do something, an attentive task, okay, something that requires attention, and so this produces a reduction. So this is the only thing that you can see visually, the synchronization of the alpha rhythms related to some task. Okay, so I mean, if you are interested, I can guide you on that even remotely, but it is a very interesting experiment to do, and it's very easy. Okay, going further with the frequencies, we have other type of bands, which are the beta bands, which is mostly coupled with the alpha bands, and is related to active thinking, but also motion planning, focusing. So it is a similar behavior to the alpha bands, and all of them, we'll see better later, are also called sensory motor rhythms, as they are the EEG rhythms related to the motor activity. The last rhythms are all the EEG signal above 32 hertz. Actually, normally it would be better to say above 40 hertz, because normally 32 is still beta for me. But in the neuroscientific literature, you find a little bit of discordance. Let's say the higher frequency rhythms are the gamma rhythms. These are not related to sensory processes, but they are really hard to measure. So this seems very useful because you can now filter your signal and associate it to each rhythms as a sort of components, but actually this is just a convention. Okay, so again, it doesn't work like that. It is a convention that the neuroscience was introduced, of course, to simplify a little bit of clustering and description of what happens at the behavioral level, but it's more complicated than this.
Yeah, my question is, because you shown the curves, and you said we can see at distance the alpha one. But usually I think, or they are usually mixed all together
Yes.
because they happen in different frequency bands. So what we see actually is always a mix of all
Yes,
these
always.
components.
It's always, but the advantage, the reason why you see this alpha is actually clearly in this slide, because when we really see what happens in the frequency content of our EEG. So for the ones of you who may not be familiar with that, this is the EEG spectrum. So that means given like a window of EEG signal, I'm trying to extract which is the power associated to each frequency. So it's frequency component, each oscillation within the EEG signal. Okay, and if you are really recording an EEG signal, you can recognize it because it has exactly this shape. So it should follow a so-called 1/f trend, which is this red one. Okay, which is like an exponential decay from the lower frequency, 0 hertz, to the higher frequency, 45, 50 hertz. Okay, now it can also go farther, but it would be so small that you wouldn't really see anything. Okay, so this is the shape that it has that follows, but actually this is the blue one is the true form of the EEG spectrum. And what you can see here is why is not going there? It's not right, no? No, I think
Direction.
yeah, it's connection. I'm disconnected from Zoom. Exactly.
We're gonna change
It's disconnected from the Wi-Fi. Yes.
the same with another phone.
This one? Yes. Sono connesso da Zoom, cioè il mio sono disconnesso da Zoom. Dai.
No, in italiano?
Otherwise you can actually put somebody else. Okay. Back again. Oh, back again. Okay, so what I was saying is that we have this peak. Okay, this is exactly the alpha peak, which is around 10 hertz. And as you can see, its amplitude is actually generally, I mean, normally you don't really consider the lower frequency because normally below 1 hertz, it's noise. And so it is removed. So if you cut your spectrum around one hertz, actually the main peak with the highest amplitude is exactly the alpha peak. So that's why you can see, because it's the important signal, the important component of your signal. That's why you can see, of course, if you concentrate, this peak is reduced, and so it disappears because it goes below this level, and you won't see it anymore. But if you are relaxed, this peak is normally very high, and you can see it clearly in your signal. The other peak that may usually appear, but not necessarily, and not in all the subjects, is a beta peak. So this is the beta rhythm, which is normally.
It's like a Zoom.
Aiuto. Okay. Doesn't seem to work.
Fais-le doucement.
Okay.
Okay.
I want to see it. Okay, so this second peak, which is the beta peak, may appear in some subjects and in certain conditions, not guaranteed to appear, but it's also an important peak in your EEG signal. And then you have all the other, so outside is 30 Hertz, there is basically the gamma territory, but we don't really want to go there because they are so tiny. There are some studies trying to work with gamma rhythms, and they can still be very informative. But it's really hard to say that what you extract here is really EEG and not something else, because it's really that small that it's basically covered by all the rest of the activity. So it can be studied, but it's really complicated. So I won't go much into this area. So I will focus mostly on the low frequency part and in particular on the sensory motor rhythms. Okay, just as a short, just to let you know that, I mean, there's not only EEG. There are also other means of recording brain activity. So EEG is not the only one. Maybe you have already seen this video, because it's quite famous and is actually already quite old, from 2012, in which they use an invasive technology for getting the signal directly inside the brain. Okay, and so normally there are two main types of systems for doing this: the electrocorticography, which places basically a grid of electrodes on top of the cortex, so below the dura mater. And so these are the user advantage that you don't have this low pass filter that I was talking about. So you're still recording the activity of multiple neurons, but now also with the high frequency. So you don't have this low pass filter introduced by our scalp and our skull. If you want to go even deeper, and I think it was the case of this paper, you can also go deeper in the brain with the multi-electrode arrays. These are needles that are placed. It's clear from this picture, which are placed exactly on the cortex and goes deeper into the cortex, and they allow now to record the signal at the neural level, so you can really see the spikes of the single neuron. So of course they allow to, as you may understand, to record brain activity is much clearer than what we'll see. So EEG activity is here. So let's say outside everything. If you go deeper, you have the ECG. If you go deeper again, you have the intracortical electrodes. They are not the only ones. There are other types of EEG recording that normally goes into the so-called field of non-invasive neuroimaging, because they are not really used to study the brain activity, but more like getting a picture of what happens in your brain. So there is, for example, the functional magnetic resonance imaging that is normally used for assessment. So to get a structural picture of your brain, but it can also, and this is normally the magnetic resonance imaging, but it can also be used in a functional way, in the sense that by analyzing how the blood flows within your brain, you can also have an understanding of what is happening in your brain, which areas you are activating, how much you are activating them, how they are connected. So they provide also a functional image of how your brain works, but it's just an image. So you only have a single picture of what is happening in your brain. The other disadvantage, as you may clearly see, is that it's not really portable, and so if you need them for your experiments, it's not that easy to get access to this. Fortunately, there is recently like a spread of this new. It's not that new technology, but now is getting much more attention than before, which is basically a portable version of the functional magnetic resonance imaging, which is the functional near infrared spectroscopy, or FNIRS. They exploit a similar principle. So the idea is still to exploit the analysis of the blood flows within the brain to understand which area of the brain are activated, but it's doing that instead of using a magnetic coil, using lights. So it is still a cap similar to a EEG cap, but now these are not electrodes. These are interleaved emitter and detectors. Emitter of lights in the near infrared that passes through the dura mater. They arrives again in the neocortex. The light propagates and scatters and goes back to the detector. The difference between the emitted light and the detected light allows to have a measurement of the oxygenation and deoxygenation within the brain. And so to have a similar imaging as the one that you would have with a fMRI, of course, with the lower resolution, lower precision, but now it's portable. So this can be also wireless, so you can be used daily, it doesn't require any means, you can just put it on and use it. So it is a very interesting technology for cognitive neuroscience in the real world. These are all based on, let's say, the blood flows, still related to the electrical activity, similar to the EEG. There is the magnetoencephalography, which uses instead the primary current, not the secondary current that I presented before, because this primary current within the neurons produces a magnetic field. This magnetic field can be captured by magnetometers. So by looking to the difference of this magnetic field, we can have a signal which is very, very similar to the EEG one, but much more clearer and less affected by noise. Why are we not always using the magnetic encephalography? Because again, it's not very portable and not very cheap. So it is very interesting. But what I just to let you know, if you end up working with the Mac, everything, almost everything that I will show in this workshop is valid also for this signal. And actually, all the libraries, all the functions, almost everything that I will show for the EEG signal, that is my expertise, can be applied also to Mac signal. So you can easily extend what I will say also to this kind of signal if you end up working with it. But again, given the disadvantages, I won't go into the details. All these different technologies have their own pros and cons. Normally, these are measured in terms of spatial resolution. So how clear picture can I have of my brain with respect to the temporal resolution? So how fast I am in recording the brain activity. And of course, the neuroimaging, such as fMRI, FNIRS, other type of tomography, they have a very nice temporal spatial resolution. So they can give a very clear picture of your brain, but the temporal resolution is not acceptable. For the fMRI is around 10 seconds. So that means I ask you to do something, to imagine something, and only after 10 seconds, I understand what you are thinking. Of course, it's not practical. With the FNIRS, things are getting better, but still we are in the order of 4 to 5 to 6 seconds to understand what is happening in the brain. Again, it's not enough. We think much faster. Okay, so it could not be okay. Of course, invasive technology has the advantage of both. So we have normally a very good spatial resolution and a very good temporal resolution. Not as good as EEG in any case, but still better. So it is clear the best signal recording ever that you can have is using invasive technology, but with consequences. They require the surgical, all the risk related to the surgical intervention, which are risks not only associated to the surgical procedure itself, but also later on, because there's always a high risk of infections, of generation of scars, and of movements of the electrons within your brain, which are not a nice thing. And it's, I think, from 10 to 14 hours of surgical procedure. And so it is not only a problem for the user itself, for its risk, but also a problem for the healthcare, because this type of intervention costs only 50,000 euros only for the device. So just a small device, which records only a very small portion of your brain, costs 50,000 euros. If you add the cost of the surgeons, the cost of the operation, normally a single implantation costs from 500,000 to 1 million, just for a single subject. So it is not practical for the healthcare system, particularly in Europe. Okay, so that's why normally it's only common in USA and maybe in China or Japan, but in Europe, I think no one is working with this technology. Maybe Switzerland, but so in all of this, in all these pros and cons, EEG is clearly the most used technology for brain recording, because it's despite all these advantages that we will see related to the noise of the signal, is still highly portable. It is low cost. It has a very, very high temporal resolution, and I would say the most important thing, it is low cost. So now there are even devices that cost less than 1,000 euros. So it means they cost less than an iPhone. but they can change the life of people with disability. So it can be really important technology to democratize brain computer interface, neurorobotics, and cognitive neuroscience. Something that you cannot do with the other technologies. Yes.
And nowadays, is it possible, like with fMRI and LM, to read thoughts?
I mean, not really installed in that sense, particularly because it's very slow. So I can read your thought in 10 seconds. So if you are like thinking something during these 10 seconds, I can only have a single image of what you are doing. So not really. It's more like a functional picture of your brain. So I can understand which area of the brain are activated and how much they are activated, and even with a bit of more processing, how they communicate with each other. So with processing, with machine learning, with everything, I can say, okay, I can somehow understand what you are thinking, but not really in functional. More impressive, I will leave you with to search a video on YouTube, because there are several. Experiments on speech decoding with invasive technology, and they are really able to decode like sentences produced by the brain, so they can really. Is an inherent speech, inner speech, and they are able to produce a sentence out of it. Again, it's not really
Efficient.
No, it works quite well. Again, it's with all the problem of invasive technology, but I would say the performance are quite impressive. But again, it's not mind reading. Is they are asking the user to think about producing the mouth movement that would generate that sentence. This is a motor activity. Motor activity can be recorded by specific areas in the brain. So what they are decoding is actually the imagination of the movement of the mouth, which is related then to generation of sounds, and the combination of generation of sounds is generated generation of words. Combination of words can be given to, I don't know, large language model to understand the sentence. So that's normally how they work. So it's not really mind reading. They are decoding the mouth activity mostly.
And do you think in maybe a few years we could do that? It would be possible?
With EEG?
Yeah.
We are trying it. We are at the very beginning. My answer is I don't know. I'm not sure, because EEG has a lot of problems, but I think we can go in that direction, at least to not achieve that results. So it is clear that I mean it is already a lose battle. There is no way we can defeat invasive technology or achieve that results. It is to extract as much as possible and then work with the application. So that let's say how we face the problem. So, but we are trying. I don't know. So at the moment, I don't know if we can do that with the EEG. Yes.
And I saw that there is a company that was working with Snapchat. It's called NextMind. And what they are doing is like they have a non-invasive headset, EEG, that will be connected to Snapchat. And when you will say yes or no, it will directly. And this is what kind of?
No, it's not speech. What they're using is modulation of the alpha wave. So if you have an electrode here, they're using the modulation of the alpha wave. So when I ask you to focus, then you will see a variation, and you associate this variation to yes. If you don't see the focus, then you associate it to no. So you have it's like a switcher. It's not really decoding yes, like yes or no. It's decoding 1 and 0, and then associate 1 to yes and 0 to no. So it's a switcher. This is something that is the most simple BCI. I mean, you can implement it. I would say, I mean, if you know a bit of Python programming, you can implement it in 1 week probably. It's very, very simple. We also have a tutorial. It's a bit more complicated because it requires real-time decoding, requires a little bit of C++, blah, blah, blah. But we have online tutorial how to implement an alpha BCI based on alpha modulation that you can use to have like a simple switcher that works exactly like that. And it's, I think, I don't know, 20 lines of code. So it's very, very simple. Not that simple to use, but simple to implement. Okay. But it's not decoding the yes, no. It's decoding just 1 and 0. But they can associate it then to right and left or to up and down. That depends again on the application. Maybe it's enough. So do we need really more? I mean, all our computer works with 1 and 0. So can we not just work with that? Since with EEG we can record 1 and 0, can we use only that for our application? But I will try maybe to say something at the very end if there is time about this.
You will not be allowed near the place they are trying to raise money. If you say in 1 week you can do what they're trying to raise millions.
I mean, because you can do that, then you can sell it very well, but then it's not sure that it works. So it still depends on a lot of. So I can place it on you, you can train, and it would work. But the point is that for working with what? So since it's very noisy and it's difficult to get, what are the applications? You may think of it. So there is space for that in the market for several applications for hub. Now the reason why there was not this much of applications on the market is mostly due to problem with the hardware. Producing the hardware for recording EEG is complicated to have them reliable and they work every time. It's complicated, but now the market is quite full of, and they cost a lot. They cost a lot. But I would say now not anymore. Particularly the GTEC company in Austria, I think they did something very interesting, which is a device. This is the device that costs less than 1,000. And it was exactly the aim to produce an EEG system that costs less than an iPhone, so that if you can afford an iPhone, okay, so you can afford a BCI. That's exactly the idea. Okay, because before it was, it's too expensive. No, now it's not more too expensive because many people want to buy an iPhone. So if they need it, they can buy a BCI, and they can do really a lot of things with even just simple devices. So probably I would say in 5 years there will be a much spreader of these systems, and so a spreader of this application that you can also build on yourself on hardware that you buy in the same way as you buy an iPhone or you buy a robot or you buy a car. You buy a BCI system and you can implement applications on that. So it is possible. I would say that maybe in less than 5 hours, in 5 years. But you see, what you can do is simple, but it still can be effective. So from a practical perspective, how do you record the EEG? Normally you have some electrodes on the scalp, and you basically record not the activity of that electrode itself, but basically the relative activity of this electrode with respect to another electrode or a set of electrodes which are called reference electrodes. These reference electrodes, we'll see it better later, are basically your zero level, and what you expect that there is no brain activity on this reference normally. And so what you measure is the difference between your electrode, your EEG electrode, and the reference. And normally you also have an electrode for the ground, which is used to collect basically all the noise and all the electrical activity that flows. The potential difference is measured by your electrode, and then all the current goes back to the ground and goes away. Okay, so this is the normal structure. Then it may depend, of course, on your EEG system. There are EEG system that has more than RFS electrode, other that has less. There are also for clinics some monopolar configurations, so configuration that use only a single electrode without any reference. But the signal they have is not that good. The best is to have a bipolar configuration, which basically you measure the EEG activity with respect to a reference electrode. Okay, here it's just an example with 2 EEG signals, one for the left part, one for the right part, and you measure the difference basically between the activity you measure at that electrode with the activity with respect to the reference. You don't normally have not only one electrode, you have multiple electrodes. There are several caps, so several configuration of channels of electrodes that you can place over the scalp. But I would say that the most common one, the most known one, is the so-called 1020 layout. The reason why it's called the 1020 is because basically we have a grid of channels which are placed homogeneously all over the scalp with a certain specific percentage from in the frontal. So this is the sagittal plane. The sagittal plane from the nasion to the ilion. So you measure basically, you have always to measure the distance from here. Then the nasion is basically this part of your between your eyebrows. Okay, and up to here, where if you touch to the back part here, you see like a concave. This concave is the ilion. Okay, so this distance measures basically the size of your brain. which is different for all of us. But the idea is that despite the size of our brains, these channels will always be placed with a certain percentage of this distance. So it will be customized for every subject. This for the sagittal plane, and this for the coronal plane. Okay, and can be also of the different amount of electrodes. You may have like smaller caps with 16 channels, with bigger caps with 32 channels, or even more with 64, and normally up to 128 channels when they cover the whole scalp. But despite that, this I think is the 64 configuration. So these are 64 channels, which is normally the most used one, as it covers enough of the brain. And the electrodes are normally named according to the brain area that they cover. So we have, for example, this F area, which is correspond exactly to the frontal lobe, okay, where FP1, FPZ, FP2 normally are on the top here, so they are above your eyes, okay, and these are on the frontal lobe. And then we move to the central area, this part. This central area is this line, this gray line here is normally related to the sensory motor cortex. Okay, so it's the primary motor area, is C, stands for central. And then we move to the back. So this is the nasion, this is the inion. We have the parietal area, which is this yellow one. And then finally back here, the occipital area. So PO or POZ or Z is the occipital area. And in green, in the lateral part, we have the temporal lobes. Okay. So the letter that you have in front of the channels defines the lobe, the area. The numbers allows you to understand if you are in the right hemisphere or left hemisphere. So if you are in the sulcus, so in the central, just in the central, they all have this Z. So FPZ, FZ, FZ, FCZ, CZ, CPZ, PZ, POZ, and OZ. They are in this central line. All the electrodes on the left will have odd numbers, increasing when you go away from the midline. So 1, 3, 5, F, 7, 9. Okay, 1, 3, 5, 7, 9. Okay. If you go on the right hemisphere, you have all even number. Again, increases. So 2, 4, 6, 8, and 10. So if I say FC6, I know that I'm around the frontal, between the frontal lobe and the primary motor area, and I'm in the right hemisphere, and also quite far. So because it's the 6. Okay, so it's a convention, but it's useful to understand at a glance where am I recording the EEG. The electrodes can be in general of different types. So there's yes.
Maybe if I can go in respect to the not the last one, but the one before that and the one before.
This one.
So no, actually the one before still. Yeah.
This one.
All right. Yeah, because you said that we usually measure the value of an electrode in accord with another one that we use as a reference. Okay, I understand with the ground. Okay, but here
no, but it's not with respect to the ground. The ground is used for the collection. It's not our zero level. So we have the ground that collects the current once it flows, then it goes to the ground and then is removed from the system. But what you measure as with the EEG is a potential difference. So it's the difference of the potential between two channels. Okay, which one is one of the channel of the EEG system. The other one is the reference. So it normally is a neutral channel, which is generally placed, for example, on a earlobe. It may depend because there are, for example, there is a company, the Neuro, that has the reference in the top part here. So in CPZ, they use CPZ. This one as a reference. So it's not necessarily a channel. So it would be ideally to have it as a neutral channel. So normally the earlobe or the mastoid, they can be used as a reference, because here for sure there is no, since there is the bone, for sure there is no EEG activity, and even in the earlobe there's no EEG activity. So that's why it's an ideal zero level. But not necessarily. It can also be another EEG channel. The reason why, for example, they use this CZ as channels is because the aim was to go for mobile EEG, so a EEG that you can use while walking. And the problem is that when you walk, you have this movement of the head, and this enters exactly in the EEG. So you see in the EEG signal, you would see in the EEG signal exactly the wave of your movement when you walk, when you move the head, your head while you walk. And since this activity is mostly present in the central part, so in the central part of the brain, if they measure the activity, for example, of FC3 with respect to CPZ, what they will have in common is exactly this way of this wave, so the ones related to walking. So by subtracting FC3 with CPZ, what I'm subtracting is exactly this common component.
Yeah, the point is actually this. I mean, depending on what electrode we use,
yes.
Actually the wave change can
Yes,
completely
there is
change.
an example because you actually re-reference it after. So you can record the EEG signal with a certain reference, but you can also change it after.
But yeah, because you can subtract
Exactly.
after. But then in the state of art, the convention, the standard is to place it, because otherwise we cannot replicate, we cannot.
It depends. It depends on the EEG cap. So it depends on the system that you buy. So there are certain systems. Normally, for example, GTEC has the default the earlobe.
Yeah.
Antneuro has a CPZ, but they allow you also to eventually change it. You can buy an additional element that allows you to change the reference electrode and place it, for example, to the earlobe. But you can choose it. But it should be said in the paper. So when you write the paper, you have to say, I recorded, I don't know, these channels reference to FCZ with ground in CPZ. I don't know. So you have to say it, you have to specify it, because if I want to replicate, I need to know which channels you recorded, where you recorded them, which is the reference and which is the ground. These are fundamental information that a reviewer will never say, will never reject your work if you don't specify channels recorded, reference, and ground.
And the ground usually is took through another electrode.
It can be normally, for example, for the GTEC, which are the ones that they know the best. The ground is FCZ. Because it's not used, it's a useless electrode, so normally we place the ground there because it's useless. And the reference is on the earlobe. For Antneuro, the ground is still in FCZ and the reference is in CPZ by default, but it can be changed. In other system, I don't know.
Can we change this after by software?
Yes, you can do it and gain back, if the system allows, the electrons will be lost. So I can get yes, I can.
Yeah, because I understand.
So I have an example exactly for re-referencing, because it's one of the things that you may be interested in your work. Okay, these electrodes can actually be of different types. Normally, we can identify three main systems. The most common one are the wet EEG. Wet EEG means that you have these electrodes, and what you do is that these electrodes has a small hole, and you insert a syringe in this small hole, and you insert some gel, which is a conductive gel, normally contains silver chloride, which is used to reduce the impedance between the electrode and the skin, and allows a better current flow. And these are the gold standard. So if you want to have the best EEG signal, you need this type of cap, because they really allow to record the best signal possible for EEG. With the disadvantage that, of course, you need the gel. So first of all, you need gel preparation, and second of all, the gel then remains in the hair, so your user then will have to shower.
And that's actually just for the small notice. We have this kind of cups downstairs, and that's why we have actually the sink and whatever. And we have also a huge pot of gel, I think.
And beware to clean the cup.
We have many.
Beware to clean the cup immediately after use very well with a toothbrush, because otherwise you risk to lose the cup at all, because then the remaining gel oxide on the electrode, then it will actually go in the other way around. So it will dramatically increase the impedance of the electrodes, and the electrodes then has to be thrown away. If it is a GTEC cup like this, it's quite easy, because you just remove the electrodes and place another one. But for example, other systems such as Antneuro have the electrodes inside the cup, So you would have to throw away the whole cup, which costs around 50,000 euros. So beware, because I was in another university in Italy, and they say, oh, they help us. I did a similar workshop on another system, and say, okay, help us put in the cup. I was not seeing anything, any signal. And I checked the cup and it was white, like full white. They never cleaned it. I said, okay, you have to throw it away. And they lost 50,000 euros like this. So please, really, you can just put it on under the water. It's not a problem. You just place the cap. Of course, not the amplifier, not the cables, but the cap. They are washable. They want to be washable. So you place it under the water current, and with a toothbrush, clean it. It's not a problem. But it must be clean. Alternative to this, so the other side to the wet electrodes are the dry contact. Dry contact are systems like this that normally are more portable. This is the cognionics from Switzerland. They do not require any means. Cognionics. They do not require any. Is the for my experience, and not that experience in dry. We normally use wet, but cognionics is the best. It's quite expensive, but it's the best dry EEG system that you can find in the market. And it also covers a lot of channels. Normally, dry EEG have only frontal channels or like a very small set of channels. This one instead covers the whole scalp, so it is very useful. They do not require any means, so they are in direct contact with the epidermis, normally using conductive pins made of gold plated. Okay, so they are really easy, simple to use because you just put them, record the data, and it's done. No preparation, nothing. Everyone can put them on themselves. They normally still requires, let's say, to go deeply in contact with the skin. So normally they have screws here, so that you have to, let's say, cut a little bit of the dead skin because they have a really high impedance. So you need to go at least a little bit under the stratum corneal. So I mean, it can be painful, but not that much. So it's still okay. The problem is that the signal is a mess. It's really a mess. So if you need a reliable EEG signal, don't go for a dry EEG system because it doesn't work. It takes all the noise, all the artifacts, everything. So don't go for it.
And eventually, because this means that the setup is complicated, but this means also that the population that you can choose. I mean, you have constraints on the population in the sense that, as instance, just something random. If we want to work with children,
it would be better to go with a dry, of course, because you don't have the preparation, you don't have to put any gel, you don't have anything. It depends on what you have to do, basically. If you, what I would suggest is that if you have to do studies, they do not require the EEG as a control signal. So, for example, you need to record the EEG signal in real time and maybe process this EEG signal. I don't know, modify the behavior of your robot or modify the behavior of your game based on what you are recording. Then with the dry, you're not able to do this because the signal is a mess. Processing it in real time is almost impossible. You don't even know what you are recording. Sometimes someone passes just behind you and you record all the signal of the foot on the ground because they produce electrostatic noise, particularly if they have plastic shoes. So it's a mess. On the other hand, for example, if you just need to record the EEG signal while you are doing your robot interactions and you want to process them afterwards, for example, to understand the level of attention, the level of effort, or some other things, then maybe a dry EEG system could be useful because it's very simple to set up, it's not intrusive, you don't risk to have like something strange happening during your experiment, and since you can work offline, there are ways to counteract all the noise and all the problems as you can. Say, manually cut the signal, manually process the signal, and in the end, end up with something useful, but not in real time. So if you need EEG for control, no dry. If you need EEG for simple studies of human robot interaction, okay, maybe you can go with dry. Actually, there is an alternative for that, which is in the middle between the two, which is quite a new technology, particularly for a system. It was mostly used in clinics, but now also for BCI, which are this called semi-dry or water-based electrodes. They use the same, almost the same cap as this one, but instead of using the gel, they're using water saline. So normally a sponge within the electrodes, and so you just have to to place the cup in this water with saline. So saline can also be the powders of for the babies. It's okay.
I used the solution saline,
Yeah, exactly.
but it was difficult when the subject has a lot of haze.
Which system have you used?
Emotive.
Okay, no,
Emotive with the task.
Yes, with the semi-drive. I would say that I also didn't consider that technology till now because they also had these kind of problems and they were almost working as a dry EEG. So I didn't like them that much. I have to say that I try this one, which is the net from Front Neuro. This is a very, very good system. So we are now buying it to use in our experiment because they almost provide the same quality as the wet, but it's super simple. You just place in the water, place it on top, and that's all. And then you just remove it, and eventually you will have your hair a little bit wet, but it's fine. They are long lasting. I use them in an experiment for 3 hours without any loss of signal, so they work very well. There are still some problems in the way, so still it's not as good as a wet, but again, it's a good compromise. So we are now moving to this system rather than this one, because this one for us is impossible. Yes.
Expensive than
No, it's the same, and actually for the Amp Neuro uses exactly the same amplifier as the wet one. So for example, you can buy a unique amplifier, which is normally the highest cost of an EEG system, because the amplifier is what it costs the most. And normally the cap costs less. So you can buy, for example, a single amplifier that works for both, and buy a wet cap, a semi-dry one, and a dry one, and they will be connected to the same amplifier without any problem. You can just swipe one amplifier and multiple caps and change the cap according to your application. But Anteura is the only one providing this solution.
My question was because I work with this with children and a lot of hairs. So when you have person with I mean curly hairs, what do you do? I mean, do you have some
Normally
some
in that case,
direction, some suggestions?
no, because I don't have that much of experience on this. So normally, for example, we have done study with children, but we started using the wet one.
So
we
you
asked
ask them to
yeah.
stay.
And we reduced the minimum number of electrodes. So we only focus on the central area because we are interested in the motor part. And so we try to reduce the number of electrodes to prepare in the minimum as possible, so that it takes like 1 minute to place all the channels and that they are good to go. Because if you have to place like 64 electrodes while the child is seated, and you have lost this tension and it's done. Okay. So again, it's a matter of
the
compromise.
space that I mean, because
With the gel, you don't have any problem. The gel, you don't have any problem. You just have to put more gel.
Okay, just
With the water, it could be problematic.
Exactly. The problem we had was that the child was so curly, then we had always
Maybe for him, it would be better to go with a wet. But there's no other solution for that. You consider that this is an unsolved problem. In GTech now exit with a very interesting technology that tries to emulate, and I would actually like to try it once on the data. And now I explain why, because they are really trying to replicate almost the same signal quality that they have with invasive using non-invasive EEG, which is the pangolin. But it requires to shave your hair completely, and they place like a grid of very tiny channels on top of your scalp that you can remove it, but is directly content is fully covering. So then by machine learning, by signal processing, by convolutions, they really achieve very interesting results. I analyzed once when I was in their company for some months. One of the data set, and you can really distinguish with their data the movement of the fingers. This is something that is super complicated with EEG, and they managed to do something similar. It's super expensive and requires to shave, but it's still better than an intervention. Okay, so let's say this is the last part for the why EEG, because you can use in all of your study related to cognitive electrophysiology, which actually goes from the psychology to the spectrum from psychology to neuroscience for studying cognitive process, functional process of the neural network, task design, data analysis, and it allows, on the one hand, maybe to improve, if you work in psychology, to improve what you can achieve with only behavioral data. You can now have quantitative data of the brain activity. And at the same time, for neuroscience, you have a direct measurement of what is happening in the neural activity. One, let's say, last type of application is the brain-computer interface, which basically allows to take this data, decode them, and use them in a control loop. I will go a little bit fast on this. This is not really part of this workshop. But basically, the idea of brain-computer interface is to use exactly what I said as an alternative communication means, to use this neural data for communicating directly to the computer device or a robot. And for several applications that can go not only for BCI, but in general for EEG, for communication and control, for motor substitution, for example, to control a wheelchair, for motor recovery in rehabilitation, and now particularly now there is a huge trend of using EEG in entertainment, not only again for controlling the device, but even just to measure the engagement, the playability, the effort, while the user is involved, for example, in a game in virtual reality. So, okay, I think we can make a break now, and then we will go to the second part.
On fait 5 minutes de pause pour la vacances. Ça va. Non, non, non. Avec tout ce qui se passe, ça va. C'est mon cas de mon thermomètre. Si, c'est vrai. Justement. Ça, c'est très bien. Je crois que la plutôt question sur encore un accident. Ah oui,
quando faccio un superamento danno sulla piattaforma e i giorni del secondo perché non si capisce le peggio di comunque pure dello sfogo, ma veramente c'è qualcosa che si fa dei gruppi che si vedono proprio mi sto a me
Vuoi venire? No, ce n'è un po' si tu veux, si tu en veux.
Pour le coup, dans ce cas,
J'en ai pas.
Non, non, désolé.
Ouais, enfin, non, c'était la matière. Marrant. Ma famille.
Mais l'autre,
En ce moment, j'ai un partiel vendredi prochain. Enfin, c'est le seul partiel qui m'en reste. Un petit peu sur une heure. Et en même temps, j'essaie d'apprendre des gens parce que je ne sais pas. Vous savez, quand j'ai du temps libre, j'en profite pour faire autre chose, je peux me former sur quelque chose qui est. Ouais. Et du coup, j'en profite de mon temps libre. D'être à la maison, voilà. Non, c'est à la maison, je peux plus. J'ai la console, je me lance dans ma vie. C'est mieux d'être à la maison, mais pour moi, je peux prendre quelque chose.
ah oui, j'avais amené ça aussi, c'est bon.
est-ce que tu trouves des clubs de nougat?
En fait, le souci, c'est que ouais, je vais essayer de faire. Mais ça va prendre énormément de temps. Et il y a quelqu'un qui fait la.
J'ai appris à créer les données. Et après le traitement, j'ai mon traitement. La suite de l'opportunité du franco.
En fait, moi j'ai repris mes études et je suis en orthophonie. Et pour mon mémoire, si ça va jusque-là, en fait, parce que c'est pas vraiment assez un peu longtemps. J'espère que ça ira jusque-là, parce que donc, mon mémoire c'est pour l'an prochain. Mais voilà, après, il y a le recrutement. J'espère que ça peut. Mais voilà, normalement, c'est prévu comme ça. Ça c'est mesurer l'activité du cerveau du patient quand il lise. On a créé de la poubelle pour ça.
Bon, ça va pas s'assurer que quand il dit à quel point c'est compliqué,
Oui, puis alors, quand il a commencé à dire qu'il allait programmer sur Python, moi, je n'ai pas du tout ingénieur.
oui, bah, moi non plus, mais une fois que, parce que je pense que vous avez donné, vous avez donné un truc, et après tu modifies, toujours plus simple de modifier quelque chose qui fonctionne déjà.
C'est ça que je crée, que d'être celui qui fait le modèle.
L'avantage, c'est que tu proposes un plein de gens qui. C'est vraiment très simple de trouver des gens qui. Est-ce que je travaille ici depuis. Le laboratoire qui appartient ici. Et du coup, bah, dans le fond, les vieux
Le gamer, tu l'as. Non, je vais avoir un. Je vais pas apporter mon PC alors qu'on va faire une migration.
Est-ce qu'il ne faut pas de mouvement
Voilà.
Quando noi facevamo il sondaggio rispetto alla riunione con i bianchi, e alla fine penso proprio che non sia stato proposto perché non ci ha detto nulla. Quindi era meglio che poi continuo a fare noi, e mandavo io la mail alla Sant'Angelo e chiedevo una riunione con qualsiasi tipo di persona abbia deciso di fare per firma. E invece, come al solito, non fanno nulla.
Et chaque frame, en fait, il va reconnaître en fait et ça va permettre
Je ne sais pas.
Une plus belle.
Ah oui, ok.
Un schéma de pour les relier. Je sais pas où elle va prendre un truc. D'accord. Donc la deuxième de la première, on va la troisième de la deuxième, et la quatrième de la troisième. Et ainsi de suite. Voilà, ça va un peu décalé. Et dès que tu arrives à 8, à 8, 9, 10, et ben, tu reviens vraiment bien. Et c'est comme ça qu'après, bah, par exemple, c'est le
No, lei che mi è stato come se
Sì, perché sono in
avec tous les fils qu'il y avait dessus. C'est la transposition de ça. Et tout ça, moi, j'ai un gros paquet comme ça. Voilà. Même si je pour moi.
Ton perso, tu l'as emporté.
Mon portable?
Ton ordinateur.
Oui.
Ouais.
Il m'avait
Non, ouais,
demandé.
ah bah, non, bah, j'ai fouillé dans la plaque. Oui, ce temps-là, après, sinon, on a
Mais,
pas.
il m'avait dit normalement, il peut utiliser dans ma tablette. Et en gros, c'est, tu vois, par exemple,
On a dit que qui la connaît pas et tout.
Bon, déjà, on va voir.
enfin,
Passer une heure de profil.
mais non, ça prend 2 minutes. Là, je te le fais tout de suite, si tu veux.
Est-ce qu'on répond?
Connecté avec ton compte Google.
Sì, mi volevo chiedere, non so se già adesso, quella review che stavo facendo, quella là. Sistematico non la riusciamo a fare, tutte le review è meglio perché qui da bere che la sistematica è un metodo molto rigoroso. Inizi da tu, io posso scrivere insomma, tutti gli articoli grazie alle citazioni contenute negli articoli. Posso iniziare a scrivere, facciamo un punto e poi iniziamo. No, quando sto qua. Poi quando finiamo, se hai tempo, no, in questi giorni sto qua.
Okay. Slightly modify the slide ordering that what you said, just to, as I said, is more an improvisation. So I just modify it so that we can go directly to a bit of a practical part, so that we don't get bored of the whole theory. So let's move to some practical things, which is we have seen, let's say, the origin of the EEG signal and its characteristics, how we can acquire it. Okay, let's suppose that we acquire our EEG signals finally. Okay, so now we need to work with them. So the very first part of the tutorial is about EEG data uploading in your program, EEG data visualization, and some pre-processing. So why we need this part? Because the problem is that, as I said, the EEG signal has a very interesting characteristics, such as working in the millisecond time scale. So it's a very a signal with a very high temporal resolution. It is cost effective. It is a direct measure of neuroactivity. So all the positive things. And then one thing that maybe was not we didn't mention that much because we will explore it better now in the with the tutorial, is that it is multidimensional. What I mean is multidimensional is that differently from other signals, there is not only a way of analyzing the EEG signal, but actually there's really different ways. For example, we have seen that surely there is a component related to time because it's a temporal signals, but also related to space because we have several electrodes, so we can not only understand what is happening in the brain, but also in the brain, but also when it is happening. We have seen also that it's not only a temporal signals, but actually the main things that we are interested are the brain oscillations. So another way of analyzing these signals is in the frequency. So we need also, we are not only analyzing in the dimension of time, in the dimension of space, but also in the dimension of frequency. Okay, so that we can understand the amplitude, how fast it's going, with the theta or beta bands. Okay, and also this oscillation, what is the main frequency, but also what is the power of this frequency, and eventually what is the phase, but we won't say much about it. The problem is that when we record the EEG signal, we are not only recording EEG signal, we are actually recording all of a set of other information that are not brain activity, which are called artifacts. Generally, these artifacts can be divided. There are many. Okay, there are really many artifacts that are recorded by the EEG system. And the main problem is normally these artifacts has an amplitude which is much higher than the EEG signal. So really finding the EEG signal among all this mess is really problematic. But generally speaking, we can define two main types of artifacts. The physiological artifacts, which are related to the specific subject in general, and the extra physiological artifacts, which are normally related to your acquisition system or your environment or your protocol. Physiological artifacts can generally be other, so not only the brain produces electrical activity, there are a lot of other biological systems that produces electrical activity. Our heart, our lungs, our muscles, they are all producing electrical activity. And this electrical activity, of course, is captured also by the same type of electrons. So we may have, and we already seen that, for example, we have the overlap with the EEG activity, the so-called electrooculography, EOG, which is basically activity of the eye. Not only the blinks, so these huge peaks that we've seen in the very first slide, which are normally related to the contraction of the muscles when you blink, but also even eye movements. So if I move my pupils to look around, then I can see this movement in the EEG signal. And it could be useful because eventually you can have also this information in your recording, but for that, maybe an eye tracker is better. Okay, so this is not, even if it is an electrophysiological signal, this is not the signal of interest for us. We are interested in the brain activity. So this for us is noise. These are nothing. Electromyography, in particular, the neck muscles. The muscles produces, for head stabilization, a lot of muscular activity that traverses through the scalp and arrives at the EEG electrodes and are recorded by the EEG system. And again, this is noise. This is something we are not interested in. And this is particularly critical in applications where you have movements. So when you work with BCI, normally you work with located patients. Located patients are still. So there's not much of blinks or muscular artifacts. But if you want to study, for example, behavioral movements, so using them with the healthy, you have to take into account that they are moving around, they are moving the head. By moving the head, you need to activate the nerve muscles, and you see the activity of the muscles in your EEG signal. Sometimes this is not really problematic for the EEG, the heartbeat. The ECG or EKG, in case of the kinematic of the heart moving, so it's not really related to the it's called EKG and not ECG because the noise is not really produced by the electrical activity of the heart, but mostly by the fact that when the blood pumps within the veins, they generate movements, no, of your arteries and veins that are contracting, and this contraction is a kinematic signal that is then it is visible in the EEG signal. Actually, not much in the EEG signal, fortunately. Mostly if you work with MEG, the magnetoencephalography, the heart rate, you see the heartbeat, you see it very clearly, and also with the apneas, of course. With the EEG is at least one of that noise that is not that problematic. But other tongue movements, if you greet, if you inspire, everything can be seen in the EEG signal, and these are artifacts. So we have to remove them. Yes.
Is it easy to remove them?
No. It is not. Just as to let you know, it was actually a combination of the two. for a European project, I had to work on a data set of walking with and without an exoskeleton with an EMG and EEG signal. It took me 1 year and a half to clean the data of 10 subjects. And I think 1 month to implement the full BCI. So the main part was cleaning the EEG, and then it took 1 month only for the BCI part, the analysis, and the publication. But 1 year and a half just for cleaning. So no, it's not easy. And the main problem is that what I will teach you is mostly my experience. There are some other tutorials, but there is not like a recipe on how to do it well. So everyone does it on his own. Everyone has his own tricks. I have my own tricks. I will try to explain, but I don't know if they are like the gold standard. There's no gold standard for EEG preprocessing. There are good and bad things that you can do, but there is no gold standard. So it depends on you, and you need to get experience by doing it.
And the cleaning always happens after you recorded the signal. You cannot add many more
yes, you can. There are some techniques for that. They don't work that well in any case. They use template matching or adaptive filtering to remove, for example. But the problem is that what you are recording on the EEG is a transformed version of the original one, so it's not that easy to do a template matching as in other scenarios. And most of the time, these techniques, in many cases, would not work in real time. And so it would make the process so slow that it would be useless then to use the EEG in real time. So most of the time, what you do in a BCI application, for example, what we do is try to work again on the application. So we take the noise, we concentrate our analysis on very specific features that we know they are not affected by noise. In case they are affected by noise, I try to counteract, for example, through the robotic behavior, through some other type of strategies. I would say that currently there are no ways of cleaning the EEG signal in real time. When we talk about EEG cleaning, I'm talking about offline analysis. When you record the data, you have your data in your computer, and you need to process the data to extract information. But in real time for BCI, it's not practical. We don't only have physiological artifacts, we also have extra physiological artifacts. The main ones is, of course, the power line noise at 50 Hz. If the EEG system is good, and if you place the EEG system correctly, theoretically, this should be very low. But it's not guaranteed, particularly if you work with dry electrodes, you will have the power line noise for sure. Okay, but there are ways of counteracting it. But not only, there can be other electromagnetic interference. For example, if you work with a robot has motorized joints. Motorized joints produce electromagnetic noise. This electromagnetic noise will go into your EEG signal, and this is particularly problematic for wearable robotics. For exoskeletons, this is my main field. There is a huge problem because every time the exoskeleton starts moving, it activates the motors, and the electrical activity of the motors gets into the EEG signal, and so I have to remove them. Movement of the wires. If you don't have a very good EEG system, or you have a lot of wires, while the wires are moving, for example, because you move your head, the movement of the wires will generate artifacts in your EEG signal. Other specific electrodes can be other specific extraphysiological artifacts, as I said, particularly for what is called mobile brain body imaging, which is the analysis of the brain activity while you move. The movement of the head, they produce very specific movement artifacts that you need to remove, and many others. So again, this is a very, very short list of the artifacts that you have to deal with, and they depend specifically on your application. So you need to identify for your specific application which are the artifacts involved and try to find the best procedure to remove them. Because again, there is no receipt, there is no standard procedure. It depends on the specific protocol, on the specific application. So we'll try to cover, let's say, the most common ones. So I will try to remove the EOG, that for sure will be present, EMG, that will be present, and the 50 Hz power line noise. So these three normally are always present. The other ones, it depends on your problem. So the aim of the preprocessing basically is to maximize the so-called signal to noise ratio. Because the problem is that, and this is actually it's not like this, and probably it's the other way around. You have, if you assume to represent your signal in form of a distributions of data, as most of the signals, they organize as a Gaussian distribution. And the problem is that what we measure is actually an overlap between the distribution of data of your real signal, which is the EEG activity, and the distribution of data that belongs to noise. We define the signal to noise ratio basically as the ratio between the amount of the power of the signal and the power of the noise. And our aim is to maximize this ratio. So normally, not either maximizing the signal, and this is normally done through the hardware level. So buying a new hardware, doing a good initial preparation will increase the signal, this part, the numerator of this part. But once you recorded the data, it's done. There's no other way. That is the signal that you recorded. And so what you can work is reducing the noise. So reducing the denominator so that you increase the overall signal to noise ratio. And you can do it in several ways. Reorganizing properly your data, so segment your data in specific times in which you are sure that that data contains brain activity and not other activities. So for the other data, the other time windows in which you are not sure what is happening, you can either remove them, say I don't consider them, or try to replace them, replacing this bad data with the correct data. No, but it's through cleaning. So cleaning is the procedure of facts removal. So that's what we will do as our first hands-on is I will show you how to upload the EEG data, visualize EEG data, and do some of preprocessing. For that, we need a bit of preparation. So that's what we will do now. What I prepare for you, I will go here. So basically what you have here is a list of Jupyter notebooks. Jupyter notebooks is basically an organization in separated shells of Python codes that we can run independently. And they need to run on a Python editor. So now as first things, we have to set up our Python editor. For that, I would suggest, but this is up to you, but if you are not familiar with working with Python or in programming, I would suggest to maybe work in groups of 2. So maybe if you are not familiar with programming, maybe work with someone that is familiar with programming so that it makes things easy. If you know how to work with it, okay, you can work on your own. Again, you will not program. I provided you with all the examples, and it's all about running the examples. But again, just to make things smoother, I would suggest to maybe organize in small groups and work together. In any case, I will show everything on my computer. So even if you are not able to run the hands-on, doesn't matter. I will show them on my PC. So
we need to extract the document.
Yes, exactly. So the first thing you need to go on the folder that you just downloaded and extract the folder. Okay, so that is like a normal folder, not an archive. And then we need to link this archive to our Python editor. You have 2 choices for that. Either if you already work with Visual Studio Code, so if you have already Visual Studio Code installed, or any Python editor that would work with Jupyter, it's fine. So I don't know, is there anyone using Visual Studio Code? Okay, so if you have Visual Studio Code, you can just open Visual Studio Code. Maybe you also have in Visual Studio Code Jupyter Notebook installed. Okay, so what you can do is go here in extensions. Okay, and in extension you can search for your desired extensions, and you need to install Jupyter Python if it's not installed, and Jupyter. So and then after that, it's going to be super easy. So you can go here and type Python. In my case, it will be already installed.
Yeah.
Okay, Python. Okay, and you install first Python, and then after that, you can go Jupyter. Okay, Jupyter Notebook support. Okay, and you can install also Jupyter. Once you have done that,
Which page you are?
No, wait. This is for the ones who are using Visual Studio Code. Then I will. There is another one. If you're using Visual Studio Code, you just you need Visual Studio Code. You need to install the extensions for Python and extensions for Jupyter. Okay, and then you're done. You just have to open the folder and it work. But I can show it. For the ones of you who don't have Jupyter node, Visual Studio Code, you can still use Jupyter online. But for that we need a little bit things to do. Which is first, you need, in any case, you need to install Python in your computer if you don't already have it. Which you can do it by here. Python download Python.
Sandro told us that we could use Google Colab.
I tried because I thought that it was possible.
Yeah.
I tried, but actually it's quite complicated to then link my virtual environment to Google Colab. So that's why Jupyter is easier. But because it's actually about of going to download Python. You can download Python for your PC. Yes.
But the virtual environment is only for the package, right?
For the package, because otherwise you need to install a lot of packages on your own.
But on Google Colab, you don't need to.
Because they are not available on Google Colab. So some of the packages that I use are very specific, so they are not available in the standard kernel of Google Colab. So you will need to install all of them one by one, and it may take a lot of time.
Okay.
And also because I provide you already with the data, and so accessing data in Google Colab can be done by uploading the data in a drive, but it takes a lot of time. Okay. So this is actually faster. So you just have to go on Python download and download Python. And follow the instructions for downloading Python. You can just walk around if you need help.
Non, c'est ça, mais quand t'as fait toujours être utile, donc autant que tu vois c'est là et que trouver une difficulté plus tard. Ce qu'on va faire, c'est j'ai vu des packages, c'est pas des packages où quand tu les utilises,
ok.
So maybe you can work together, yes. Because, yeah, it's going to be possible. I think yours is a bit too old.
Là, c'est notre dataset, c'est là où on aura les données qu'on va utiliser pour entraîner l'IA. Et donc, juste faire la visualisation pour voir à quoi ressemblent les données. On va devoir les nettoyer, je pense, avec
Yes, perfect. It is on Twitter.
vous les aviez déjà.
You just have to open the folder. Yes,
je l'extrait.
then I will show you to select the camera. Okay, then you're good to go. Thank you. Yes. No, you need to start the whole the whole
Visual code. Comme c'est dans le
Yeah, I think so.
Mais normalement, c'est voilà,
Celui-ci?
You already installed Jupyter and Python? Yeah. Once you install the folder, you can just open the folder.
Il y avait bien
Okay. So in your case,
il va tourner en arrière.
you already installed the Python and Jupyter extensions.
Python,
C'est pour ça que j'avais
J'utilise même, on peut utiliser.
Yes, so you just have to open the folder. It's already unzipped.
C'est par rapport à votre ordinateur, vous l'utilisez pour une notification telle, donc c'est bon.
D'accord.
Okay, just open.
Est-ce que ça peut tomber?
Okay, so
Ouais.
who's still installing?
Ensuite, c'est pas du tout.
Who's not using
Oui,
Visual Studio
mais
Code?
c'est pour le.
You're using
No, I'm not.
you're using just Python.
Yeah.
No Visual Studio Code. Okay, perfect. It's already installed.
Yeah, we did. You should need them number on.
Yeah, now I will show it. Sorry.
On va faire ça.
Ah, it's not on the
Déjà, on
side.
va utiliser autant.
Anche Python non ho problemi, diciamo,
Ma però la cosa è più complicata da usare.
ok, come vuoi. Il y a quelqu'un qui est en train de l'installer qui a des soucis, qui veut de l'aide.
Non, parce que déjà, tu nous as induit en erreur.
Anaconda si usa, Anaconda separare, Anaconda separare. Si c'est vrai que Google Colab, je pensais que c'était bien. Et l'idée aussi, que vous pourrez travailler en binôme, ok? Donc, si vous voulez vous organiser en binôme, ok?
Yeah.
Vous voulez travailler en binôme, mais peut-être multidisciplinaire. Alors, c'est bon.
On
Ok.
travaille en binôme.
D'accord, ok, c'est que Anaconda, donc c'est très bien, ok. Là, ok. Peut-être être un pro, toi. Un pro, toi.
Lei lavora con me perché non lo può scaricare.
E tu l'hai installato?
Sì, sì, fatto
Oh, che
Il
bello.
a
Tu peux retourner dans
Oui, je retourne.
Il y a l'ordinateur qui va installer là. Et comme ça, on pourra ça va marcher. Oui, en fait, quand l'installation, en général, on l'installe dans un endroit qui est connu. Mais en fait, par défaut, il ne faut pas le voir.
Mais avec non, ça va prendre du temps pour extraire.
Non.
Oui, vous êtes en train d'extraire. Ouais, pareil, ça prend du temps après.
Il se fait mal.
The whole folder takes a lot of time.
C'est quand on peut
Maybe it's because of the data set. If the data is a lot of
Let's wait. You should get fast as soon as you possible.
just we can go here Python.
J'ai ramené des trucs, mais c'est pas suffisant. Donc, j'allais chercher un complément.
Oh non, ce n'est pas.
No. Just okay. Who else? You was installing Python? No, you're using Visual Studio Code. You are installing Python. Let me just check.
Si je peux le redémarrer.
j'ai repris mes études en orthophonie.
En master,
Là, je suis en master.
master 1.
Master 1. Je crois que c'était le choix.
Mais s'il y a des trucs à enregistrer de votre part.
Redémarrer, c'est ça que tu veux faire?
Quand on le met dans le chemin, il faut le redémarrer pour que l'ordinateur nous mette c'est donc là quand on va le faire. Moi, je suis en école d'ingénieur et je suis en dernière année, donc je suis en équivalent L2. Et moi, je suis en intelligence artificielle et data science. Et je suis en train de faire des études dans le laboratoire. Grâce à une IA générée, sur la normale avec des recettes, des parcours,
Mais c'est ça, exactement. Je sais pas, sì, adesso possiamo cioè. No, scusami, dove avevi usato che volevamo quando dobbiamo finire? Ok.
Et les gens ne sont pas vraiment. Oui,
C'est des conseils en fait.
mais j'avoue que j'ai pas, moi, je ne suis pas renseigné. Moi, je fais plus que la génération de la production, ce n'est pas encore. Même pas comment on dit.
No, ma anche fuori dal c'è una storia buona mezz'oretta.
Mais en fait, il fallait.
Sì, 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 vai.
Oh, okay. So one thing first to ensure, because I don't know why it's taking very much time on unzipping. So if you have still this .zip, you extract all, because it may take a while to extract everything. I don't know if someone of you already did it. So is anyone the folder not already unzipped? Does anyone of you already have the folder unzipped? Yes, sir. It worked for you? Okay. So for all the others, unzip it, and then as soon as it's ready, I will let you activate it. But so in the meanwhile, we can go on. I can show you basically the same thing here, and then as soon as you're ready, you can go. You have a Visual Studio Code?
We are.
And maybe for the others who don't have the folder zipped already, maybe you can group with someone else who already has it, and then we can switch. No, who's ready? You are ready. You have already? No? Who's ready?
I'm ready.
Maybe you're ready?
We are already at the next
Okay.
We tried. Je sais pas, Python library.
Any word?
We don't know, we just discovered the file.
You already on it? No, not
Je suis
yet.
déjà dessus, donc et j'attends. En attendant qu'on arrive à faire 5 ans.
So meanwhile, you can work with you. And maybe you can no, not yet. Okay. Just in the meantime, either you split with the rest while it's unzipping, or you can just follow me on my computer. Okay, so if you are working with Visual Studio Code, as probably you are already, okay. If you open the folder here, okay, so you should have something like this, okay. Let's open the first notebook, which is data visualization and processing. So double click on it and we'll open, and you will see something similar to this. Okay, so now on the top right corner, you can click here. Okay, it will allow you to select the environment, which is the ones where to run it, and you will see there is one suggested, which is .venv Python something, which should be inside here. So .venv scripts Python X, and click on that.
I don't have this.
You don't
Yup.
have,
Okay.
we have a Python tool.
So, question start.
Python environment. Okay.
Instead of this.
Ouais, moi, c'est des systèmes.
Just select one.
Ultra rapide.
This one, okay.
The one here, bon, quand j'avais mis le chemin, j'avais mis mauvais, donc là, je l'ai remis dans un sandwich encore redémarrer, mais on après, on a
We have the same.
alors, tu vas prendre. Donc, comme je disais, en fait, il a pour utiliser fichier Python en général, on doit importer des fonctions autres. Et ces fonctions-là, en général, on doit les installer. Donc, c'est pas que. Et en fait, ce qui est bien, c'est qu'il nous a donné.
And if we follow the instruction.
Il nous a donné son environnement avec les trucs déjà installés. Et là, on a juste ajouté les trucs. Et normalement, quand on appuie sur select le kernel, on est censé pouvoir le voir et dire, ok, moi je veux celui qui m'a donné. Et là, on ne voit pas. Donc,
It requires
un petit peu.
Ça marche chez vous? Non, c'est pas.
Il faut un package apparemment supplémentaire.
Bah ouais.
Oui.
Est-ce que tu sais pourquoi mon VS Code il voit pas les trucs du dossier, genre le V11? Il ne le propose pas.
Il ne le voit pas à gauche?
Bah c'est il y est là, dans les dossiers.
Là, tu le vois là-bas, mais là, il ne
Là-bas, il
le
ne le
propose pas.
propose pas.
Non, moi aussi, il ne me le propose pas, mais je crois que c'est juste parce que normalement, il est censé voir le python.exe qui est à l'intérieur.
Ok.
Et soit quand on lui a donné l'accès au dossier, il n'a pas accès au sous-dossier. Peut-être c'est pour ça. Enfin,
Ça, tu sais pas le régler. Moi, VS Code, j'utilise jamais, il donne des boutons.
Moi, je peux faire. On peut exprimer le kernel. Ça peut être sur le côté.
Normalement, il est censé donc le voir. Là, parce qu'il y a donc le dossier là. Il ne voit pas. Alors, pardon, pardon. Il prend mal. Mais c'est juste on a une dossier. Oui, il est censé le voir celui-là. Là, je ne sais pas pourquoi il le voit.
Mais le même truc.
C'est pas un truc chelou, on dirait qu'on leur dit de posséder.
Ça marche.
Bon.
Oui,
en fait, il va nous réinstaller on essaie de l'avoir déjà installé. Mais si vous voulez encore, moi je suis là, mais je trouve de connecter à l'autre. Votre compte Google, Microsoft.
Moi, il y avait un truc où ils ont dit,
LUTIN service. Extraire les dossiers online.
le truc, il avait dit,
Ouais, c'est juste pareil, en fait, j'ai pas accès au.
Faut choisir parmi ceux-là.
Ah, c'est bon? C'est bon. Ah, Enter, Enter, maintenant, c'est tu peux aller dans le data. Non. Tu vois, le fichier, il est pas encore là.
Il y a peut-être des trucs avec
bah, ça, c'était dans les derniers cours avec la
une forme de.
Dans mon ordinateur, remplir le code.
Ah, oui.
Why do you store right? Just here?
No,
Or you downloaded it.
yeah, we don't.
Que faire avec ton document?
You have to pay Non, ça.
C'est sur ma chose que je veux.
Ah, c'est bon. Ouais, tu copies le lien de Python pour les langues. Après, tu as un petit truc comme ça pour aller dans. Non, pas celui-là, mais qui est tout en bas à gauche. Non, un tout en bas à gauche. Un setting. D'accord. Après, tu écris kernel ici.
Quelle version?
Maintenant, le choix, c'est de prendre un bouton et puis le code. OK. Et je pense maintenant que tu fermes le BSC, tu le relances et tu devras pouvoir le trouver. Mais quand tu cliques dessus,
Sur l'environnement?
c'est mieux ça. Après, c'est parce que tu n'as pas compris peut-être.
Ouais, déjà.
Non, ici, dans le.
Mais alors, non, mais en gros, ça m'a fait un truc comme quoi j'avais fait ça.
Non, mais l'extension, c'est juste pour qu'il le voit.
Je me propose que.
Et il installe.
Ouais.
Mais ouais, et du coup, j'ai dû cliqué sur installer Python, ça m'a envoyé sur le store installer Python. Et
Ouais.
puis ça marche chez quelqu'un.
Non, moi, ça marche pas.
Ah,
Pour l'instant, le digital est toujours encore. Peut-être que c'est beaucoup plus bon,
ouais. Ok. Pas d'idée.
I don't know.
Ah,
Close to open very good. No. Let's see. Whoa. Okay, let's do this one. Instead of the way I need to do this.
Alors, je ne dis pas que je fasse ça.
Je sais pas si vraiment ça se dure, et dès qu'il fait des tâches un peu plus vite d'installation, il ne veut pas. Bah, le marqueur, il dit, pas du monde à la personne, ça.
Okay. Okay. Now. Where is the folder? There is yeah, okay.
Oui, comme ça. Hein? Non, voilà, non, il va terminer.
Okay.
Non. Il était très près de...
Ils avaient mis ça en place l'année dernière, avant, il y avait là. Parce qu'on avait passé de matériel pour tous les questionnaires électroniques.
Alors, c'est des skills dont tes profs ça a administré.
Non. J'en ai eu de la non, parce que la neuroscience, l'université. Après, j'ai
Tried it several times. I don't know why it doesn't work.
De rapidité. C'est logique, tu sais que les gens dans tous les sens. Question. Mais j'ai pas réussi à m'installer. Ça marche. Ça, j'ai rassuré.
Ça marche.
Non.
Et vous le voyez ou pas le.
Bah, en fait, non. Enfin, je sais pas si, je sais pas trop ce qu'on fait.
Ah, vous le faites avec Visual Studio ou vous le faites avec.
Visuel.
Ah oui, Visual
de
Code.
code.
Je pense qu'on va devoir le faire avec le CMD.
Might work.
Et je fais avec le CMD, mais ça marche pas non plus.
Même avec le CMD.
C'est la première film de
Vous avez appuyé sur le bouton dans le champ. Ça marchait, oui. Là, c'est là qu'on fait plus.
Là, on a peut-être besoin pour notre
sélectionneur aussi. Ah, ça, non, le truc est bien, ça les laisser par exemple.
Ah, oui, ça marche. Donc maintenant, il faut. Afficher la slide où il met les.
Il est là, maintenant, mais il ne rectifie pas.
Et en fait, quand on voit le code, machin, il propose devant, là. Donc,
Qu'est-ce que c'est?
Tu vas là, tu vas select
Ouais.
Python environment. Ouais, Python environment. En troisième.
Bien, c'est sûr.
Normalement, là, il devrait reconnaître.
Oui, c'est ça.
On est donc ici, mais après, pour le vrai, quand tu cliques sur le run, ça ne va pas grand-chose. Le code que... C'est un clavier allemand. L'ordi, je l'ai eu quand je travaillais au Luxembourg, et là-bas, ils ont
Et vous l'avez
Avec une machine.
C'est pas nous qu'il faut faire, c'est lui.
En même temps, peut-être qu'on exécute cette première ligne.
Moi, ah, ok,
Mais
ok.
sans le dollar.
Sans dollar.
Principal, Jupiter, oui.
En commande, là?
Oui, et vous juste vous collez et vous lancez. C'est bon, là, je suis en train de faire.
C'est le truc, d'accord.
Il y a la virgule. Ouais, en attendant, mais je sais pas, avec cette interface, c'est que pour le montrer, vous avez une identité, comment ça s'appelle, éclairée de ce qu'il a envoyé.
D'accord.
Mais vous ne l'avez pas écrit, non?
Je les ai quand même copiés encore.
En fait, c'est le plus long, c'est de le copier. Comment je le fais faire même maintenant, on a pas.
De toute façon, là maintenant, on fait la pause. Parce que tout le monde est en train de crever déjà. Et après, au fur et à mesure, on pourra voir un peu ça pour faire partir un peu tout le machin. OK.
Ben, tu lui demandes quand même, parce que
Si,
je
non,
pense
non.
qu'il veut y arriver avant.
Stefano, vogliamo mettere tutto in pausa così si va a mangiare, poi così ci accertiamo anche come si fa. Ok, on commence la pause et on se voit. A lei funziona.
Mais en fait, moi j'avais réussi, mais c'est parce que j'avais du mal à ouvrir le folder.
Sì, pas des autorités.
Ok. Resolution processing. Sì, bisogna installare. Ah, non so se non semplice. Questo non semplice, se installa il problema. Sì, ma non lo fa. Ça, c'est juste une solution. Elle te dit non,
l'environnement
oui,
que
je
j'ai
sais
mis
bien.
pour kerner Jupiter. Le kerner Jupiter,
Bah,
c'est juste l'outil qui va utiliser l'énergie.
non, non, ce sont deux choses différentes. Ça, c'est la manette.
Non,
C'est
c'est pour
le
faire la
contrôle
critique.
bloc.
Déjà,
C'est pas.
conseiller exciter pour une
Ça, c'est-
Donc, du coup, moi, je fais ça.
Je peux pas les utiliser quand ils sont
Si, ma ici, il te dit.
La connexion dans le
marche
fichier,
mieux,
en
dedans.
archive,
Si
on
tu
peut pas
veux.
encore les désarchiver. Peut-être l'archive, c'est un moyen de rendre le fichier moins lourd. Quand on a plus de le déplacer.
Tu sei più avanti.
Ah, ok, però comunque c'è.
È giusto.
À la fin de l'environnement,
Essaye de
tu
faire
demandes si tu veux utiliser le genre et après celui qui est
MN, la bibliothèque, ça va tout installer. Alors, moi, je sais pas.
Mais moi,
Je sais pas si c'est.
Ah, tiens.
genre là, installé. Est-ce qu'il faut celui-là Voilà.
Non. Attends, clique là.
Le bas
Est-ce que
cycle,
tu
c'est
l'as
pas
installé? Je te l'envoie. Attends.
Et pourquoi il me le met pas là?
Bon, ok, reviens pour l'installer.
Il y a tellement de trucs installés.
Sorry, something wrong.
Vabbè, te dico un secondo. Reviens, mais est-ce que c'est quoi tout ça? Donc,
Nous, on a juste compris ça et on a compris l'extension.
ok, oui, clique. Parce que ces extensions-là, tu les installes dans les environnements virtuels. Mais non, l'environnement virtuel. Non, ça c'est une méthode déjà, la suggestion qui t'auto-complète toute seule. Juste une chose, fais un dièse aussi sur un important moment. Ok? Un dièse, vas-y. Non, ça marche. Juste pour comprendre. Ah non, ok, alors les versions de branche, ok. Installer tous les. Attends. Oui, ces bibliothèques qui sont liées.
On va pas utiliser le code, je prends des calculs.
Oui, mais ça.
On va utiliser le terminal de base. C'est juste une interface, mais je crois que c'est la même. Mais je crois que je peux l'afficher comme ça pour le code de base, c'est quand même.
Non, les filles, attends, instant. Je pense que c'est pas ça. Il y a quelqu'un qui connaît comment utiliser qu'est-ce que c'est ça? Visual Studio Code. Tout à l'heure, utiliser Visual Studio Code.
Moi, j'utilise en général.
Oui, ah, tu aussi, tu dis. Comment on installe des choses sur un environnement virtuel? Parce que moi, normalement, je le fais avec un Anaconda.
Moi, je fais avec Conda.
Mais après, tu as
Mais c'est pareil, en
une
fait.
console devant.
Ah, et c'est où la contrôle?
Elle est en bas, normalement.
Ah bon?
Bah, elle est là.
Ah, tiens.
Alors, on me dit,
Et donc, et qu'est-ce que j'utilise? Pipe?
Bah, c'est une conférence, oui. Mais ça, je crois qu'il l'a déjà fait.
voilà.
Et je ne sais pas qu'est-ce que vous avez fait.
Et je m'en ai non
Oh,
plus. C'est pas nous.
Je vais juste me connecter au code, parce que
senti, per installare la cosa utilizzi pip o...
Però non uso Anaconda io.
Ok, con... tu hai Anaconda, sì, qu'est-ce que tu as installé? Qu'est-ce que tu as installé? Anaconda... Mais il y a,
Alors, là, je sais pas c'est quoi. C'est très chaudrine. Là, je mange c'est très spectral. Ça fait. Oui, voilà. Donc là, on va faire. On va regarder.
Là, j'ai un carnet, mais c'est pas celui.
Ouais, c'est pas le même.
J'ai le 3.11.5, par exemple, mais c'est pas un vent. Je ne sais pas si ça compte.
Non, c'est pas le même. C'est pas prendre
Ouais. Ouais, parce que j'ai trop dans le machin.
Vous avez Python d'installer sur votre ordinateur ou pas?
aussi. Oui.
Est-ce que quand tu ouvres un terminal, tu peux accéder à Python depuis ton terminal? On va voir. Bah, sinon, tu fais, au lieu de faire avec Visual Studio Code, tu fais ce qu'il a écrit dans la slide et ça a marché.
Mais c'est ce qu'il a fait, mais ça marche pas. Ça marche non.
mais non, ça marche.
Non, mais maintenant, pip, ça marche, mais il y a autre chose qui marche pas. J'ai pas compris ce que c'était, mais ça va juste qui marche. Ça va plus, c'était sans compétence.
Oui, un endroit où on peut
Parce que
rentrer.
là, à deux, on a les compétences d'une personne normale. Moi, je pense pas que je suis pour 0,5.
Alors, en fait, vous êtes en train d'installer tous les packages normalement.
Si, MML ou pas, et ça fait plus là.
Mais le monsieur il a dit qu'il y a des packages que l'on ne peut pas installer via LUTIN par moment. Donc, à un moment, il va être bloqué. Voilà, c'est bon.
Déjà là,
C'est mieux.
ça allait.
Tu vois.
Déjà.
Là, si je mets ça,
Voilà.
ça me. Ça crache.
Bam.
Et on va. Bon, là, j'ai un peu de temps.
Bam, bam, bim, bam, boum, bam, 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 bim, bam, bam, bam.
There is no people selling. Ah bah, si je
Alors, moi, est-ce que, ah, c'était comme ça déjà, on l'a fait.
Non, là, ça marche pas. Écoutez, c'est bientôt que ça marche, à un moment que ça va plus marcher. Mais...
It works.
It works?
Comment on
ouais,
peut d'abord
Tu installes pas et puis tu installes à main.
Yeah, they didn't use your environment.
You're installing from scratch.
Yeah.
And maybe I don't know why, because she's actually able to access my environment. But then when he enters inside, he still didn't see anything. I don't know why. It's exactly the same procedure that I follow online.
Yeah, we download the CMP. And then it's on my
Mais du coup, je t'en étais sorti pour non.
I was checking what was the best file, but you can go back.
Oui, c'est des étapes en fait.
Toujours la même chose.
Here and it should work.
Le travail que j'ai et le
Sorry?
travail.
So you installed all the libraries?
No, I use your
And yours is working.
yeah, I
À cause
use
d'avoir une proportion à un peu pour chaque, par exemple, accident. Là, tu as un pour chaque électrode. Non, c'est le grand bordel.
En fait, les étapes après, dans le premier, tu veux les refaire. En fait, à midi, il n'y a pas vraiment de mémoire. Il y a des chercheurs. Voilà, il y a des chercheurs qui vont suivre les étapes d'une certaine manière. Il y en a d'autres qui vont inverser certaines étapes. Mais en fait, on peut transparent sur les étapes et sur les étapes. C'est sûr, les étapes, c'est pas. C'est encore pire. Enregistrer.
Chez nous, on travaille. Moi, je vois, on travaille.
Oui, mais je pense que c'est l'institution que je savais en fait avec la mission. En vrai, oui, c'est logique, tu vois, mais il faut intégrer une activité. Mais c'est là, c'est vrai. C'est bon, c'est chaud quand même.
Mais là, tu parles des corolles.
Tu as des problèmes avec j'ai compris. En gros, c'est
un miracle. Il faut vraiment
un environnement où il y a déjà les packages installés qui est dedans, dans le point VEND.
Et
Non, on est juste censé l'activer.
même
On a déjà les
fait
packages
les
activés.
choses bien.
Sauf que la même, on est installé comme il est.
Et
Et on
on
est
arrive à voir
dans
le
l'environnement qui nous dit qu'on n'a pas les packages. C'est pour ça que tout le monde est en train de réinstaller.
Mais nous, on est habitué à travailler avec. Dans la Covid, il y avait des gens. Ouais, mais il y a les mains, on sont tous des honneurs, on est. Oh.
Ouais.
Mais
Le
dans
noyau
le champ
nord
des neurosciences,
plus,
c'est pas comme ça. En fait,
c'est ce qui va exécuter.
c'est comme là, c'est sûr, on sait, voilà.
Non, alors, l'effet est que si tout le monde essaye, va obtenir plus ou moins la même chose, c'est normal qu'après on sait.
Non, mais justement, là, ils ont sorti des affaires sur ah ouais, c'est changé. Je sais pas que les deux contre un qui ne pas se trouver sur le jeu, mais c'était des tâches. Je peux rien, je
Avec des lances. Attends, c'est plutôt environnement.
mais
là, là-bas.
oui, mais c'est genre vraiment des trucs qui considérés comme
Et donc, il y a.
c'est sûr sur telle zone et telle qualité.
Plus le premier pour voir.
Ouais,
Voilà.
chose.
Ouais, c'est pas censé simplement un.
Moi,
Comme tous les chats.
j'essaye de minimiser les dégâts. Après, Stéphane, ah ben, écoute, bon, il y a toujours. Alors, ça, c'est. Il faut pas. Juste une chose. Vu qu'il y en a entre nous, ça, c'est la punaise des choses qui m'arrivent tous les temps.
Ouais.
Ok, chaque fois que j'organise des ateliers, des travaux pratiques avec Python, moi, je me prépare toutes les conneries, etc. Faire tout le bordel. Donc, ce que vous êtes en train de voir, c'est plutôt normal. Même un jour, si vous allez à un proposeur de TP ici, là, ça pourra vous arriver. Vous vous trouverez dans des situations comme ça. Donc, vous ne stressez pas. Ok. Non, un futur, si vous allez à faire des TP, vous ne stressez pas. C'est bon. Ça fait. Non, non, c'est bon. Après, on arrive à résoudre. On essaie toujours d'essayer de minimiser les dégâts, mais ça.
Ça fonctionne.
Ça fonctionne.
Le following selected par EEG. Ah non, mais c'est juste pour le faire en fait.
C'est bon.
Non,
Il a tout à fait.
Ah, tiens, addirittura.
Tu as besoin d'avoir des données assez rapidement.
Et t'as pris
combien?
Je suis trop. J'ai compris la phase de vente à l'hôpital. Après, j'ai été là, j'ai été électronoseur. Avec vendre, là ils disent choisis
Ah, mais ça, c'est ok. Ça, c'est ce qui est. Et moi, à ce point, j'ai ouvert les filières et j'installe des choses.
En fait,
Marc Portier, ben non, pareil.
on a calculé la distance entre.
Mais du coup, là, c'est ça.
Je ne sais pas, peut-être qu'il y a une manière plus élégante de le faire.
Si te dit comme quoi il n'y a pas de...
Senti, io quello che ho fatto è stato di infilargli qua, di dirgli di puntare questo qua. Poi giustamente non funziona perché non ha installato le cose. Allora,
Ou plutôt autour des branches. Comme ça, du coup, ça
e poi anche un paio, ma
Dovrebbe essere già installato.
dovrebbe essere dentro di questa environment.
In assoluto lei per esempio non vede neanche il Python interno, io però non so perché. Cioè lui lo vede, c'è Python.
on a réussi, par magie. En fait, en gros, il faut que tu installes toutes les extensions, les librairies qui sont inscrites. Là, c'est dans le code.
Bien, bien.
Ouais. Et en fait, on va t'installer LE,
So, when I was at the show,
c'est pas comment il a fait.
Dai, secondo me, no, celui-là.
La magie.
on the terminal,
Il a téléchargé
Pista, la
install map
via le terminal en fait directement.
Le personnage,
Il va me fermer. Et puis,
Moi aussi, là, je viens de réussir à lancer le programme, mais j'ai dû réinstaller le
fallo una volta. Va. Après tu fais MN et tu fais un
Salvo.
non, mais tu fais la feuille géo, il va te mettre les derniers qui
Non, mais
ouais, c'est plutôt ça.
ah, perché non facciamo con est-ce que tu peux le
Computer is very
Okay, to the
tu
same
t'installes MNE.
oh,
it's finished, do the same for MNE, then do the same for Mumbai should normally be installed.
Okay.
Then you should work.
Mat,
So you're not using everything.
Ok.
But it's mine, yes, I prepared everything.
Et-
And you can use directly this one without installing anything, but
Et ensuite, je prends quoi, ça résout le problème.
I don't know why.
J'espère que c'est nécessaire, mais.
l'explication. Après, quand on run, ça va.
Ah, c'est ce qui. Ah, mais en fait, du coup, il n'y a pas son.
Non.
Je sais pas pourquoi c'est.
Ouais, je pense que lui, ça marche. Il avait déjà des trucs installés en fait.
des construits entre plusieurs organes
io ho levato anche la togliese, questo qua. I deleted
Because for, then if you delete the first one, then you don't have interactive plots.
Ah, that's why. Okay,
That's why
okay.
you know there is another package that I installed it. I don't remember. It is Pi 2 T5 installer Pi 2 T5.
maybe
Instead of what?
No, now let's finish first and then I
Okay.
will tell you. Then if it works with both of you, then we move to all the rest.
It's
I think my
It really works.
I
I'm using my environment.
no, I reinstalled I create another
But you could select the others, they did the same, but it was not working. I don't know why for you it was. Let except it was set up. So I don't know why it's not going for you.
restarted
Okay, so you're the for you. I don't know why.
Je croyais que
et cliquer et
Non, parce que. Non, non,
supprimer le banc et oui, j'ai tout validé.
Ça marche, mais sont des images non interactives. Là, avec ça, tu as des plots interactifs. Ah,
C'est ce que j'ai aimé, il m'a montré.
exactement.
Pourquoi ça marche?
Parce qu'on installe des petits écrans.
Ah, ça n'a pas marché directement.
Parce que toi, oui.
Non, mais je sais.
Et j'ai des trucs qui se téléchargent, des plots, des machins.
Tout lancé en même temps.
Ah ouais, ouais, ouais. J'ai l'ordi qui est sur les rotules, il est chaud comme ça y va, quoi. Faudrait peut-être que je l'arrête.
Tiens.
Ah, moi, oh, okay, for you it was. Yes. Okay, okay. And you just installed everything.
Also the thing, yes.
It works. For you, it's working with my
Okay, for you, it's working with your
I don't know why only for him. It's
He
working.
installed it, yeah.
Yeah.
Maybe there was some incompatibility with the end.
Probably. I don't know why.
Ah ouais.
En fait, là, si on run all comme a fait Jérémy, on va aussi avoir des.
Ouais.
Ah non, mais pas j'ai.
Faut le faire au moins une fois, histoire de.
Ah ouais.
Ah ouais, d'accord, j'avais pas vu.
Oui, ben, oui. Ah, ok. Tu peux le faire même en fois. Ok, amuse-toi.
Ça marche. Ok. Bon, on va aller.
Je pense que cette deuxième partie de régistration est utile, non? Parce qu'il y a, on écoute juste le bruit de vous qui Donc, j'arrête là. Je pense que, parce que je peux enregistrer, exactement.
Bah,
Mais je pense que je coupe la première partie jusqu'à la pause, parce que je pense que c'est la partie intéressante, non? Ok, et après, après, on verra, je vais mettre en ligne, on trouvera, peut-être qu'on va faire un canal du temps, et on va mettre tout là comme on fait. Ok. Okay.
On aura déjà les